Good morning. Good morning, everybody. Thank you for being here. We weren't expecting this crowd, so I'm really glad that you're joining us today. My name is Silvia Zuffi, and I'm welcoming you to the CV4Animals workshop, Computer Vision for Animal Behavior Tracking and Modeling. So actually, I'm welcoming you on behalf of the whole organizing committee. That is Alge, Yusuf Park, Sarah Berry, Anju Kanazawa, Shohei Nobuhara here, and Anna Adamansky. So this is the third workshop, CV4Animals workshop. We started in 2021. And I would like to take a chance to actually recognize the work of the... You cannot... Oh, yeah, sure, of course. Can speak closer. Can you hear me if I speak like this? Okay, great. So I was saying that in 2021, this group of people decided to suggest having a workshop dedicated to animals at CVPR.
Recording in progress.
And that was a great idea because so they set up a community that is growing year by year, as we see today. So, but where did we start as computer vision for animals? Many people like to think that the first person that attempted using imaging technology for observing animals was Edward Muybridge. And in the 19th century, he was wondering if a horse, when running, has an instant where all the legs are in the air. And that was not possible to detect by simply looking at a horse running at a high speed. So he designed and implemented an acquisition system where he captured video. And this is a frame that demonstrated his hypothesis. And this is a nice example of early ways for exploiting technology to observe and understand animals, which is computer vision. But the story of computer vision applied to animals has not been always successful, because at the very beginning, with early attempts, when the computer vision model was too simple, actually using automated techniques was kind of reducing the richness, the complexity of the task that one could perform. For example, in studying animal behavior, people were using a very simple model to represent a rodent with the tip of the nose and a center of the body. Only very simple actions could be represented. But we are seeing recently, actually from a few years ago, that now we have very, very effective and generic trackers for landmarks, for example. And with these new tools, we're setting the ground for understanding complex behavior. But this is just the beginning, and we know how the animal kingdom is huge. The kind of questions we are interested in are super complicated, and there are many challenges here. We see a few of them, like complex camera and subject motion, interaction and occlusion, complex environments, in particular the deep sea. And I'm very, very, very 3 times excited to be here today and to have the opportunity to discuss these challenges with you and see your amazing work through your oral and poster presentations. And now I'm giving the stage to Helge that will give some more details about the workshop. Thank you.
Yeah, welcome everyone from my end too, and yeah, feel free to come in, find a seat on the floor. I want to have a picture for the organizers showing how many we are working on computer vision for animals. So hopefully next time we get a bigger room. When organizing these workshops, it's always a little, will we fill the room? This time I wasn't concerned at all that we will fill this room. So yeah, great that you all came. I'm glad to see that there's lots of interest. In fact, I have some stats for you from the last years. We have a pretty stable number of papers, number of submissions. And that's nice to see. We have always something like 30 submissions, which is quite a lot for a workshop, I would say. So yeah, head out to the poster session later on, check out this great work. We only could fit so many orals in here. And also last year we tried this journal track. So we collaborated with the IJCV journal with a special issue because this workshop was intended to provide a platform for us to publish as well, to collaborate and chat, but also to publish. So the workshop is good for dissemination for in-person discussion, but the journal is really good if you want to place your work somewhere and really have it visible. So last year we had a completely open journal track. We were kind of overwhelmed with the number of submissions. Maybe some of you were a little disappointed with how long the review took. We too. So we refined the process by now. So this year we have an open call. We selected the best papers from the workshop for submitting to the journal. We invited them. And also, if you haven't submitted to the journal, but you want to be part of the IJCV special issue, then send us a 4-page abstract by July 10, and we'll consider you still for the special issue. And the final deadline is August 33rd for submission of the IJCV. And yeah, reach out to us if you have any questions concerning that. But yeah, so let's move on to the workshop of today. We have a nice lineup of speakers. So Kostas will start with the keynote. Then Tanya will join afterwards. We have our oral session, 2 poster sessions in total, one right before the lunch. You will have to move all the way over to the other building, but that's also where the lunch is served. So head over at 11 to the poster session and smoothly transition to your lunch break. And then, yeah, we continue in the afternoon with, again, 3 keynotes, another oral session, coffee break, a panel discussion, don't miss this, and finally the poster session. So this is also all on our webpage in case you lose track. Yeah, this is just the path, right? Here's the big cruise ship, so exit the building and towards the other one to find the posters, and then also find your way back to us in the afternoon. And we only lately got the poster board. So on the webpage, and if you have a poster, check out what your poster ID is. It starts with 100 to 120. And yeah, put up your poster just when the poster session starts. So I think a little earlier would be possible too, and take it down after. Yeah, so this is how the workshop's going to look like. I don't want to take much more time from the presenters. We want to start in time. But one last thing, I want to thank the program committee. It's really a pleasure to organize a workshop if the reviews come in time, if you find enough reviews. So thanks a lot to a lot of you reviewing the papers which are submitted and giving feedback to the authors how to improve. So thanks a lot to you. And yeah, without further ado, we can move on to the keynote, I think.
Sure. Should I take off the cable from this laptop?
Yes, you can share it.
Oh, okay. But how I'm gonna see my note? Can I see my notes still? Yeah, I'm on Zoom.
Single screen then.
Okay.
Yeah, maybe we can take the HDMI cable.
Yeah.
Then you can.
Where's your Zoom application?
Yes, yes.
Oh, yeah, okay.
But yeah, if you want to see your
I
slide
would like
notes,
to see my slide notes. Yeah.
Oh,
Yeah,
yeah.
let's try with the HDMI. Do you have a
Yeah.
direct HDMI cable or need an adapter?
Old professor cannot do talk without notes.
Can you start the Zoom? Can you simply start the presentation with a presenter tool as you want?
So I take the HDMI, right?
No, that is possible.
It's not possible.
It's also
Yeah, I have done it.
Okay.
not now. And can you host?
Good idea. Can you hear me? So I will introduce our first speaker while we are taking care of the technical issue. So first of all, a warm welcome also for me. We are very excited to see you. We are excited to have the first keynote
It's okay
speaker,
to take the, I can take
Professor
the
Kostas Danilidis, Professor of Computer and
the-
Information
the-
Science at the
The
University of
visitors
Pennsylvania.
can see your screen.
He has made numerous contributions on visual odometry, omnidirectional vision, 3D pose estimation, 3D registration, hand-eye calibration, image matching, and his interest today is in geometric deep learning, data association, event-based cameras, and vision-based manipulation and navigation. So welcome, Kostas, and we are thrilled to have you.
You should have the presenters do in the here, I think.
here, right?
The original PowerPoint.
He's a magician. So you can do Zoom without projecting the screen and still see your notes. Oh, people on the waiting room. This is Helge's. Right.
Good to go. Thank you.
Great. Thank you so much for first for organizing this for one more time and for inviting me, Shohei, Anna, Kyunshu, Helge, Sylvie. When we started this work, actually, I knew only Sylvie's work. And now it is really an amazing community. I'm going to present very recent work on tracking that appeared at IJCV 2 months ago. And this is collaboration with a neuroscientist, Mark Schmidt, and work from Xiting Zhao, Yufu Wang, Amon Berndt, and Mark. And I will give also a brief historical perspective. First, happy Father's Day to all the fathers here. We are going to talk about families in the aviary environment. So computational ethology is really a very exciting field. It is the challenge of capturing the social interactions of animals in complex environments outside the lab. And there's a very nice article actually about the importance of emerging technologies for computational ethology by Ian Kazin and Connor Haynes. And they say that a rigorous and predictive science of behavior will likely prove essential for the development of effective conservation strategies and policies in the face of increasingly rapid anthropogenic changes, meaning also climate change. I would do this research even if it would be completely useless. I'm a big proponent of advancement of useless research. There is a book, The Amazing Usefulness of Useless Knowledge, from Albrecht Fischler. Yeah, that is a challenge. Yeah, that is yours or me? Oh, okay. Sorry. And the big advancements are advancing because of many fundamental technologies and discoveries that have been done before we even knew that they were useful. So, see if the videos play. The videos are much nicer usually. And we built in 2017 and 18, we built the Penn Aviary at the University of Pennsylvania, where we, yes. Optimized for video, correct. Yeah. Optimized for video.
Maybe if you, I think you would have just stopped sharing.
Oh no. Go again. You Zoom people, you owe us something here.
Do you want to share the sound?
Yeah.
Yeah.
I think there is some singing there. All right. So we have constructed an aviary where we have installed 24 cameras and 8 cameras and 24 microphones. And these were to study cowbirds. Cowbirds are singbirds. And we have known that singing has been one of the main mechanisms for mating and reproduction. It is one of the biggest instruments of evolution to promote species. And originally, my collaborator, Mark Smith, together with David White, they had studied how the birds interact if you remove the parts of the brain doing a real ablation, not what we call ablation in deep learning. I mean, deep learning, every study is called an ablation, even if you don't remove anything. And so they indeed removed a part of the brain, of the female brain, which corresponds to the song. And they have seen that the social organization of the whole colony of birds changes completely. And we want to decide this without ablations, but ultimately by observing the birds with cameras and microphones, and also putting a neural recorder in the brain of the birds. So this is the Penn aviary, which was constructed by my postdoc, theoretical physicist Bernd Frommer, and Amon Perkis, PhD student in biology. This is outside with natural illumination. You see the April tags that we use for continuous calibration of the cameras using our own system called TaxLam. And the cowbirds are caught out in nature. It's very easy to catch them in North America. And we have a continuous population of about 15 cowbirds. So we have a full three-dimensional, we are able to do a three-dimensional registration, both of sound and cameras. And this is why I show only a static snapshot, because this is what we have been doing for a while the main result here will be really the dynamics of the birds. And we really want to understand the moment-to-moment behavioral changes over several months, in particularly during the mating period in the spring. As you see, if you have seen other setups, which are mainly indoors with very controlled backgrounds and illuminations, this is extremely challenging environment. My students keep telling me that we really make everything very hard and that I don't learn anything from 2,500 CVPR papers. So the first thing that we know by talking to biologists is that 3D pose matters. So the birds are not only singing, but actually they are doing also several changes in their pose in order to indicate their intent. And this pose is worth to find per se, but also it's worth also because it's helping in track.
There is a lot of
trajectories. There has been a lot of advances with Deep LabCat and Sleep, Deep Fly 3D, Alpha Tracker, the Monkey Lab from Kyushu, which actually many of them are also very well replicated. Neuroscience at Penn has full installation now. And this is the most recent and related to our approach. This is the iMappet from Constance, where tracking is done by tracking semantic key points. And there is an associated, actually also paper, the 3D POP, which is a paper at CVPR. I counted at least 3 animal papers at CVPR. We are doing very well. So we started originally with a Penn aviary dataset, where we did a single shot processing. And when I say single shot, I really mean that when I say shot, it is really 8 viewpoints from the same time instance. So we annotated 6,000 instance masks in order to train a RCNN dataset. And we also annotated several semantic key points in 5,000 instances. And we also have annotated specific semantic 12 semantic key points, which we used as additional actual information in loss functions for reconstruction. We also established a full actually mesh model, as you see here. And we have run a full optimization using the key points, as you see here, and the silhouettes as, I don't know why it is advancing by itself.
On our side, the Wi-Fi is too weak. We should switch that for the next talk.
No, something is wrong with my talk as well. But we optimize both the reprojection of the key points and the reprojection of the silhouettes on several views. And we got quite satisfying results from that, which we published at CV20 and CVPR 21. But the main challenge for the biologists is not just get the individual pose, which they could use also, but really being able to find for every actually bird where it has been every single time instant and whether it sank or not. So we started collecting a tracking dataset, being motivated by actually annotations of behavior, like male sings, female chatters, male moves closer, female flies away, male chases, and using 10 cameras and 40 frames per second. We concentrated on 3 behaviors that I will show a little later in the slides. And we clicked only the initial position and the final position during a track. We didn't really click all the masks that we have detected. So this resulted in about 720,000 clicks per minute times 12 hours over, which were 4 days per month, times 3 months. It became 51.8 million clicks, and you're going to see a lot of people acknowledged at the end. So as you see again here, we emphasize what will happen if we cannot, can I just switch the projection? Oh.
On Zoom is fine.
On Zoom is fine? Oh, okay. But not here. Anyway, so we called it "Where Did It Land?" Dataset, the wild dataset, because we annotate only the start and the end of the points, as you see here. When they sit on a perch, and where they end up being on a perch. And we have 987 such motion segments, which we have split in training and testing. So this is how it looks. So the where it starts and where it ends. So we mark from green to red. And this is from several views, from 4 views from the same sequence. So the competition here in terms of data sets, as I said, is the 3D POP data set that will be presented here and at CVPR, which is based on semantic key points and also additional ground truth by motion tracking. I don't know if you have used also human. I think there was some human annotation, this semi-automated. Our tracking pipeline looks as follows. We both use the trained masks, the bird detection mask from our previous data set, but we use also a motion detector based on a Gaussian mixture model. Then we triangulate the masks into point clouds. This triangulation happens actually by using the trifocal constraint, where for every pixel, not using any key points, for every pixel, we choose triplets of views and we combine the triplets, and we do it only for about 20 pixels inside the mask, so it's not a combinatorial explosion. We can check with the trifocal constraint whether 3 pixels are really geometrically consistent. Then I will show how we track over shorter and longer tracklets. And in addition, if we want, we can perform an re-ID and then analyze, have some initial analysis of the social behavior. It's very funny because the analysis of social behavior has been done more from my group. I'm not even actually, my collaborators from neuroscience are really laughing, but it's quite interesting. So we started with the MASCAR CNN detections look like this. In the original MASCAR CNN, we had quite a good success, like almost 80%. Then we take the motion segmentation masks and we combine them through the trifocal constraints to the point clouds, which have been, you see here, the reprojections. We run connected components to get the point clusters. And then we do 2 things, both of them based on papers by Ouellet's group, both from 2006 and from 2019. First, we regard every reconstructed point more like a random particle because it doesn't always correspond to this. There is no way with the uniform texture and the lower resolution that these birds appear. There's no way that this is really the projection of the same point. It's just the closest point. So pretty much you have a point cloud that is randomly moving with a lot of noise because it doesn't always correspond to the same surface point, but still has about the same motion direction, unless, but only locally because the birds are actually flying when this is happening. So the wings are always moving. Still though, because from the velocity constraints, we can somehow track, and we're going to see that this is actually one of the biggest obstacles, the constraints we have in the individual small tracklets. Then we take the 3D tracklets and we resolve them by projecting them back and forth in time so that we find a consistency in the motion. And this is how we establish longer 3D tracks. So this is on the top, this is how several actually instances from birds while flying. You see the variety of the poses. This is a very challenging actually appearance compared even to articulated mammals or birds which are just walking. And you have a 56 frames sequence here. It is a short sequence which has been actually tracked quite well. You can see actually the tracks from 2 different viewpoints and individual instances with the red dots. The red dots are becoming bigger when the bird is coming closer to this corresponding view. And the bottom is a trajectory with 300, I think, 75 frames, which has been combined out of 3 tracklets. So the results are not good. I mean, no, I'm honest. I'm really honest on that. 10%, this is 10 centimeters accuracy. So this is ours. This is the stereo matching method from Wellett's group, which is really only reconstructing stereo points. And we, what they inside, this is for 10 centimeters, 30 centimeters, 50 centimeters, and 1 meter. And this is how many are detected within that accuracy as correct. And we see that we actually, even at the 1 meter, our in terms of when it is less than 100 frames, we detect it by 75%, but only 44% within 10 centimeters. Between 100 and 300, it is 30% within 10 centimeters and 61% within 1 meters. And for bigger than 300, it is only 16% and 44%. And this is when we have this is where it improves significantly, where we use a re-ID oracle, which tells us at the final position whether what is the ID, and if it is consistent the ID, then actually we say that this is the same actually tracklet. So we can still work. So why I say it's not good? Because you cannot, in tennis, you take the Hawkeye and you give it to the tennis world, and the tennis world has the Hawkeye as a god, right? I mean, this is the ground truth. Here, you cannot take this system and give it to a social scientist and say, don't do anything else. This is what I mean, they're not good. For computer vision, for these conditions, I think I am extremely happy, and it's a very good start. And we can still do studies with the correct ones of them, because we know, based on this experiment, which were correct. So the main challenges are, how much time do I have? 5 minutes, okay, yeah, I will do that. So the main challenge is the variation in flight shape. You're gonna see it here. And please remember it when you compare it with other, remember the challenge that we have, the challenge that we have here. We have a lot of shadows, and with also a lot of complex backgrounds. You see even people running behind here. I mean, we don't detect them as birds, but and we have 15 birds at the same time with very many mutual occlusions. We are going to still work on the tracking, obviously, and I will show what we plan to do. But the tracking challenges that we face are also tracking challenges in multi-object data set. It is due to occlusion that we merge the point clouds, that many times because of the wing somehow is having completely orthogonal direction that does not it doesn't get into the same point cloud. Here, although this bird flew like this, that this is detected as 2 different birds when they cross, and the shadow as well. And there are several challenges. I'm gonna not go through this, but we have them in the paper. There are instances of incomplete detection, of split detections, a lot of challenges because of occlusion, and a lot of challenges because of variation in the lighting conditions and the appearance. So, but we already can study a few things, and this has been already used by the biologists. When we combine the 3D pose plus the tracking, the people find specific behavior. For example, when actually specific bird, because we know its ID, is always doing this, like when the angle between the beak and the neck is exceeding specific angle. We have run also the re-identification with ResNet 50, and the only hope that we have in this re-identification is because if you have seen the beginning, we have put little rings of this color, which is like blue, purple, blue, teal, purple, green in 2 different legs in females and males here. And these are the detections. These are the detections by re-identification. This is what we used in combination with the tracking in the table below. Now, something very interesting to close it in a more like a social note. So based on that, we studied 3 behaviors: approaching of the male, singing of the male is always the orange here, approaching of the male, singing of the male, and the female is leaving. Okay, and here we have the frequency of these approaches. You see here that specific females that are, this is the first action. So this means the blue here means that this happened many times. The teal purple female approached a male, and they approached the female, which was BTF. This is like the this male here approached the red yellow female. This means that this female left and this female left too. And you can see specific behaviors that this male, for example, is always leaving when others may leave, chasing other males, not females. And these are actually, these frozen actually frames here, they show these approaches in real life. And we use a definition of bonding, like when they really start, when there's a bond, which is a definition from the biology literature. And this is when a female receives more than 50% of songs from the single male during like a whole hour. So singing, 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 half of the time you sing this, you listen to the same person. And we have the first action as approach, leaving, and singing with a probability here. And we have a probability of no pair bond. And because many probabilities have the same, so the male singing to a female and the female stays with the male have the same probability actually of pairing and not pairing. But if you subtract the probabilities, the most successful strategy that is usually following by a male is when the female leaves the male and then later the male approaches the female. Remember that. So this has been presented in the Helge was the associate editor. Thank you. It is public at HACV, and this is you can see all the tables and the pictures that I have shown. And we started capturing birds with event-based cameras. Guillermo is there, maybe other event-based people. And the main key about event cameras is these are each frame shown here is only 5 milliseconds, not 25 milliseconds that was in the previous cameras. And we really see that it is really a full continuity. And even with a very simple connected component and Kalman filter, and here we plot the centroid, which is not, we don't do the Kalman filter on the, we don't impose a lot of smoothness in the centroid. We can still actually, we can already get some much more promising results than with the regular cameras. So let me summarize the, so 3D shape pose matters, but makes tracking much harder when the birds really change their poses. We have to deal with complex flight trajectories from viewpoints that are not like a bird's eye viewpoint, like in mouse environments, for example, and with very challenging backgrounds and illuminations. So what we learned, we definitely need to optimize the camera placement. We need faster cameras. We started experimenting with RFIDs, which can really actually tiny. You can really put in the birds even without a backpack. And really to expand tracking, not only in this environment, which I find it already being wild. And I cannot, I mean, 3 times actually, the birds, actually there was a disease and it is really for the biologist, the life of a biologist student who has to go and feed the birds every day and clean actually everything. It's really much harder. And but we still, it is worth expanding the tracking in the wild by combining both high definition, like in space, RGB, and faster event-based cameras. And remember, female leaves, male approaches is the more successful strategy. And I want UFO and Gini has done 90% of the work in tracking. Professor Vijay Balasubramanian, which is more from theoretical and statistical physics, is studying now the social interactions. Mark Schmidt initiated everything. And you see other Nikos, famous from the 3D humans work, and a lot of undergraduate students who really collected data and annotated them. And thank you again for organizing this workshop and inviting me. Yes. Yes, yes. So for the birds itself, it was done using supervisedly. Yeah, for the masks. For the key points and the tracklets, it was done all by hands without any automation. Yeah. So I mean, we need still. We definitely need more data for the tracks, but I strongly believe on the event-based cameras that we will avoid annotating more. That would be a great idea. I mean, they're not, they're indeed very energy efficient. One could wear them actually on a helmet with a very small battery. With them, we still want to capture simultaneously color. So the main challenge is more to build a good beam splitter to capture the same image rather than creating a stereo pair between events and event camera and RGB camera.
I have missed it, but how do you detect when a bird is singing?
Oh, yeah, I haven't presented this. We have a full algorithm for sound localization, and this also has been annotated. But we have a full algorithm for sound localization. We have a one-off commercial system. We have 24 microphones, yeah, which are all synchronized with the cameras. 24 of them. Oh, the April tags. The April tags are used for the calibration of the cameras. And actually, we also calibrate the microphones by moving a sound source with April tags.
A question on the re-identification problem. So have you explored even localizing the rings or some kind of
Yeah.
ring?
That's a very good question. So the rings, the problem is that most of the time they are missing because the birds eat them. So, we tried a lot actually. That's why we never try to click on them or, but maybe with higher definition views. This view is not from our cameras. No, this view is not. This is from a professional videographer. These are hardly visible, but still, actually, we have studied in the ReID in the paper. We study in how many views they are visible. Again, by having students who said yes or no, and how the tracking helped.
Yeah, related to this question, did you try with extracting features from texture?
Yeah. The ReID is running with the feature from the using features from the last from a layer from ResNet, because the ReID is not running as we run a separate ID classifier, which is like fine categorization classifier. It's not we don't run ResNet with 15 categories. We take the features frozen from the ResNet and we run 15 like the way we do face recognition with contrasted triplets as well.
Yeah.
Thank you very much.
Thank you, everybody.
Right.
So, Tanya, you can share your screen, and in the meantime, I'm giving an introduction.
This should be shared. Hopefully you can see and hear me.
Yes, thank you, and we hear you. So I'm really glad that Tanya Berger-Wolf can be with us remotely, well, even if remotely today, because she's very well known by the community, but I'm giving anyways an introduction. So Tanya is the director of the Translational Data Analytics Institute and a professor of computer science engineering, electrical and computer engineering, as well as evolution, ecology, and organism biology at the Ohio State University. Her research is at the intersection of computer science, wildlife biology, and social sciences. And Tanya is also a director, a co-founder of the Artificial Intelligence for Wildlife Conservation software nonprofit WildMe, home of the Wild Book project that most of you, I'm sure, know very well, which brings together computer vision, crowdsourcing, and conservation. And Wild Book has been featured in media, including Forbes, National Geographic, the New York Times, and CNN. So prior to going to SEU, in 2020. Tanya was at the University of Illinois at Chicago, and she holds a PhD in computer science from the University of Illinois at Urbana-Champaign, and has received several awards for her research and mentoring, including the highest honor of the University of Illinois, UIC Distinguished Researcher of the Year, U.S. National Science Foundation Career, Association for Women in Science Chicago Innovator, and the UIC Mentor of the Year. So this went very smoothly. Thank you, Tanya, for being with us, and the stage is yours.
Thank you very much thank you everyone for organizing the workshop, and in its third year, yay, going strong, and for inviting me. And so I'm happy to be able to join you from an undisclosed location at an Airbnb in Chicago, Illinois. Because of the bureaucracy, the State Department's bureaucracy, I cannot unfortunately interact with all of you in person. Happy to follow up in any medium that's available. So with that, I would like to introduce imageomics. Some of you already have seen some versions of this introduction. Happy to share new results, new directions. So we, scientists and biologists and many and humans in general have observed nature and derived insight from visual observations for centuries, with increasingly more sophisticated technology for visual observations that allowed for increasingly sort of expanding set of questions and insights about nature. Technological developments allowed us to zoom in to the atomic level or to expand to planetary scales, still providing that ability to derive insight from visual observations. Then came genomics, which changed quite a bit of what we were able to look at literally or kind of digitally, and what kind of insights we were able to derive. And for the first time, we were able, with the sort of development of genomics, we were able to connect phenotype to genotype, the appearance and the collection of traits to the underlying genomic foundations in many different ways, from genome-wide association studies that allow us sort of to understand the connection of the variation in the genome to the diseases, human and animal, for example, the phenotype of diseases. The comparative genomics provided insights into evolution and development, and more recently, we can even manipulate the phenotype by manipulating the genotype through CRISPR. And so that became one of the central, really, sort of quests in biology, understanding the connection between phenotype and genotype. So with the further development of technology, we're able to look literally or figuratively at more and more aspects at a higher resolution and higher scale of the natural world. The recent paper led by David Stuy, who is at an adjacent workshop at CVPR leading the Earth Vision workshop, but with the names recognizable by this community, many of the names recognizable by this community. There is a paper in Nature Communications in 22 that outlines a lot of the approaches of machine learning for conservation, but also overlaps with quite a bit of just general foundational science of understanding the natural world and ecology particularly. So this technology ranges from satellite and autonomous vehicles to on animals' body sensors, in situ sensors such as camera traps and acoustic sensors, and the crowdsourced social media platforms and citizen science contributions through these devices that we all carry. CVPR is a prime example of the advances in the the methodology that allows us to take advantage of the primary data type that comes out of many of these sensors, which is images. Images by far the most abundant and readily available source of information about the natural world. And with that, are we able now, are we at the point where we can leverage that technology with the methodology, with advances, computational advances in machine learning and computer vision to really start connecting phenotype to genotype from images directly. So that means not only to be able to take these millions, and it is truly millions of images coming from all of these varieties of inputs, and be able to say, okay, this is the species of bird, which we can do right now reasonably reliably, depends on the bird, depends on the image, depends on the context. There's a lot of depends, but still, we at least know how to do it, and when it goes wrong, what we need, what we need to change. But can we go beyond and say this is the species of bird because it has a yellow belly, black behind the beak, and it wobbles when it walks? How do we get there? So, and what are these things, the yellow belly, black behind the beak, and wobbling while walking? So those are biological traits. Traits, as a quick reminder, are characteristics of an organism that are describing its physiology, morphology, health, life history, demographic status as in female, male, juvenile, adult, and behavior. So this can be beak color, as I said, stripe pattern, fin curvature, fused teeth, scarring, size at birth, pollen feeding, nest weaving, or even things like an opossum playing dead when threatened. They started, many of you may be familiar with the traits that started with a study by Gregor Mendel on peas, and that's the size and shape of the flower, the pea, the pod, the plant itself, and so on and so forth. So it's the traits themselves are determined by genes, the environment, especially behavioral traits, and the interactions among them. And so what we want to do and what we need to do in order to connect phenotype to genotype is really to make traits computable, preferably directly from images. And this is the promise of imageomics. We envision it as a new interdisciplinary scientific field focused on understanding biology of organisms, particularly the biological traits and observable phenotype directly from images. This is founded by the National Science Foundation's Harnessing Data Revolution Institute grant. The core, it quickly expanded partners. So we have here a large group of people where the community is growing, and the Costas' talk, previous talk, is a great example of, you know, what we envision imageomics to be is this biology directly from images, photos, or videos. And I hope that the examples that I will show in the next few slides really speak to you and sort of particularly to this community, and we can collaborate to expand the range of biological discovery in our understanding of the natural world. So the approach that why we think it's really feasible to get right now, we're the good point to get to the because part, and really to understand biology and infer new biological traits, new insight, because we can there is now, as I said, an abundance of image data about the natural world coming from many different sources. There's also a good and increasing connected set of other types of data, other modularities of data, which is text coming from descriptions of images or field guides. There is geospatial data. There is the molecular data that quite often is connected to the image, and directly or indirectly. And then so we can start with that and leverage the structures, the biological structures, which are, biology is the science of structures. This is where the taxonomy and the taxonomic approaches, started. This is where a lot of ontological approaches, the ontologies, building ontology structures started. So biology has a variety of structures that we can leverage to constrain and guide the architecture of the neural networks and other machine learning approaches, so that the outcome, if not explainable, is at least interpretable, and really pointing to potentially new traits. So with that, it's a great idea, we think. Does it actually hold any promise? So let's try. Let's try to get to this. This is this species because. So we tried with fish, not with birds, species classification, leveraging this idea that species that share taxonomic concepts such as common genus. So this is the taxonomy itself, not the phylogeny, have similar traits and hence hopefully likely to express similar features in the neural network layers. Let's see. Let's compare. So this is a taxonomy of a group of species of minnows, about 200 species of minnows. The closely related species are hard to distinguish, even for specialists, for experts. And so the idea then, we can try the basic thing possible, the basic approach. If we start with just the species classification without the taxonomy versus species classification with the taxonomy, are we going to do better when we add the taxonomy information, sort of in the hierarchical way? The answer is yes, it does, particularly when you're in the sparse data regime. So, okay, fantastic. Not only it helps when you're in the sparse, so in the zero and few shot learning, but and when your data isn't perfect and kind of there is a little bit of a distribution shift, it really helps in those instances. It also, so then the next question is, so can we infer new biological traits? So can we do it by constraining the latent space with phylogeny, right? The embedding space with phylogeny. And so this is, the answer is also yes, and this is the paper by Mohana El-Hamoud and a large team of Imagenomics team that will be the first oral presentation here and also accepted to KDD 23. We essentially infer the imageome directly from images by leveraging the phylogeny structure, the phylogenetic tree that has sort of the deep history of evolution encoded in it, not just the taxonomy. So you can see more details at the talk that's coming up. So the question is, why do we think that computational approaches can tell us more than what we can observe with our eyes? So first of all, our hardware, the visual human hardware, is not perfect. We do not see everything that is seeable. We don't even see traits that are biologically meaningful. There's a paper about a year ago, or half a year ago, that showed that the color variations that are visible for predators are not in moths, are not visible for humans because we don't have enough acuity in the red-orange spectrum. Not a shocking discovery once you think about it for a little while, but it is visible and easily distinguishable with computational approaches. So there are traits there to discover when with the help of computational approaches that humans are completely oblivious to. And so with that, we another project, Imagenomics project, is a classic example of mimicry. This is two species of butterflies, Heliconius butterflies. This is Heliconia serrato. All of these red columns are the same species of butterflies. They're just the variations are in different, sort of across the range in different habitats, but they are the same species and easily interbreed with each other. These yellow columns are the Heliconians melpomene, another species that are all of these, the ones within the species are more closely related to each other than they are to the ones that look like them. So this melpomene is mimicking, mostly melpomene is mimicking Errato, the yellow columns mimicking the red columns, to gain the protection, because Errato doesn't taste good to the birds, and so melpomene is mimicking Errato to appear the bad tasting butterfly. And so again, the question here is, from the visual appearance, can we tell what is similar enough in appearance, which traits make them similar enough for the birds, but yet distinguishing enough between the species so that the butterflies from the same species mate with the right ones, right? So they don't make mistakes. And so again, the first question, is there a signal? And using the just species classification with triplet loss, the answer is yes, there is a signal that in the embedding space, with the distance in the embedding space between butterflies of the same species, so this is the Errato, melpomene, is much closer than between pairs, non-mimic pairs of different species, and the mimic pairs are in between. And this is sort of variations across different triplet loss approaches, and sort of the answer is consistently yes. We can distinguish the same species versus not the same species, and the mimics are somewhere in between. Great. So what about butterflies and birds? Does it change when you have in their models of butterfly vision and bird vision for the right species? So they differ also within species, across species. And so we took the models of bird acuity, not full vision model, just acuity, and applied it, and butterfly acuity, and applied it to the images. And again, asking, is the accuracy, right? Is the accuracy for recognizing the same species versus mimics, is it different between the butterflies and the birds? And how does it compare to the original? So the answer is that when we train the classifier just, let's say, on Errato, and apply it to melpomene, the difference, so the birds, or train just on melpomene applied to Errato, right? The birds are more confused, right? The accuracy of recognizing the accuracy of the difference and of recognizing the mimics on mimic pairs is much, versus recognizing conspecifics is lower than the butterfly one. So butterflies, they really do that. They do not see mimics as the same species. This is the take-home message, right? And we're continuing with this study to understand now, placing landmarks, biological landmarks on the butterfly wings to really understand which part of the image is most salient for the birds and for butterflies. Sort of this is starting with the transformer models, just to taking a walk in the latent space of seeing what are the parts of the butterfly that are different between the co-mimic species. Here's one co-mimic pair here, and then we will ask the birds directly, hopefully by printing these butterfly, the minimum set of butterflies with the features that should be sufficient for the birds to recognize as mimics. and see versus not, and see which ones the birds will pick on most. So another imageomics project, again focusing on this idea of what are the traits that are necessary that we can infer from images, focuses on the individual level traits. So the butterfly is a species. This is individual. So we can recognize, and Wildbook, some of you have heard, so is leverages that ability to recognize individual animals from images using biometric features such as zebra stripes or leopard spots or the shape of a whale's fluke or dorsal fin of a dolphin. And so with that, then one of the questions we get, particularly from kids, when we tell them that we can recognize individual zebras, they're like, oh, a baby zebra stripe similar to its mom's. Great question. The problem is, again, human ability to do that falls short in order to be able to provide the answer. Because while we're really, really good, and there are good evolutionary reasons for it, at comparing faces and quantifying similarity of faces, not only in humans and primates as well, and even in seals and other animals, particularly forward-facing animals. The baby looks like its mom or the baby looks like its dad. We cannot do that for zebra stripes, for example. So if I asked you, is the top pair more similar to each other than the bottom pair, we cannot learn the ability to answer this question. We have no way. We have no middleware, right? No infrastructure to answer this question. But because we can recognize individuals and because we can sort of evaluate the similarity of patterns using computer vision, we can now take the information on the relatedness of animals and compare it to the similarity of the patterns as quantified by any computer vision approach. And so again, we started with the question, is there a signal? The answer on a data set of Indian leopards, where we have the mother-child information, so not the father and not the genetics in this case, just the mother-child, mother-cub. And the answer is for the facial images, can we, the way we asked it, given an image of an animal, who is in the most similar one in the data set other than itself? And so here, the pairs of a cub on the left, mother on the right, that was pulled up for each other as the most similar. And the highlighted areas are the ones that algorithmically were determined. This is using SIFT features, the most similar aspects, why the algorithm made this determination. So the biologists are collecting more images. We're also focusing on, there's in collaboration with Jan Rubenstein, who's part of Imageomic, biologist from Princeton, collecting data set on zebras, including genetics. So we will be able to directly correlate. And we are working with biologists who study eggs and egg patterns, where there are better data sets where we can more directly answer these questions. So watch this place. There already work that's starting, that hopefully will start to show up soon for public. And finally, following up on Kostas's talk, can we infer behavioral traits from video? And can we do it really directly in the wild? So there, the structures that I used are these behavioral ethograms that are complex, hierarchical, but occasionally direct cyclic graphs kind of structures of behavior. And as we saw in the previous talk, with sufficient annotation, we can sort of kind of maybe. So we had to, in this case, baboons in the wild in Kenya and at Impala, had to simplify the set of behavior quite a bit to start answering this question. So this is direct video behavior classification of baboons, and you can see their errors and sort of missed behaviors, particularly the false negatives. We took that baseline, and so over the last year, we've been teaching an experiential introduction to imageomics course to graduate students from ecology, biology, computer science across all our partner institutions, and opened it up to a wider set of institutions to participate. And we're going to do it again, and starting in the fall 2024 and going through 2024, 2025. We invite you to send your students to join the course. So over the autumn semester, a team from 5 universities, of instructors from 5 universities in computer science and biology, provide background in both the necessary biology, ecology, computer vision, machine learning, and broader computational approaches. And then in January, we take the students to a field site, in this case in Kenya, at Impala Research Center for 3 weeks. Throw them in the field, tell them go and do something amazing. They inevitably do. This year it was 5 projects, ranging from understanding the ecology at the bottom right there, the ecology of fish in Kenya, to top right is the evolution and the development of the toe pads of geckos. And then 3 projects focused on inferring behavior from videos. And in this case, video is drone videos. From that took a little while also to figure out, and this is a whole other talk of how do you plan drone missions so that to get the best data for animal behavior. But from this drone from the drone video, we extract the behavior, sort of the mini scenes, then annotate behavior. And the first pass, the first approach, and the first question was, there are traditional ways that biologists do behavior observations in the field. Do these compare to the drones? Can we capture everything that that biologists would normally see through behavioral observations? Can we capture all of these with drone videos? And the answer is yes. The drone videos, what we can see from the drone videos is a superset of 2 types of typical behavior observations. If you don't plan the drone mission correctly, you can see the most common behavior is going to be vigilance, head up, and then run. But once you plan the mission, you can get other types of behavior. We had to simplify behavior set quite a bit. The advantage here of these particular animals that we can recognize them individually. So both giraffes and zebras, 2 species of zebras, they're individually recognizable. So we didn't need to put little rings on their feet. This can be done reasonably uninvasively once you figure out the drone. And we're making the data set available. The big question here is not only to be able to recognize individual behavior, but to be able to recognize the social behavior, just as Kostas was talking about, in the wild, and interspecies interactions, as you're about to see here. So this is between hartebeest and zebras, and then watch as giraffe passes through. This is a beautiful example of interspecies interactions. So there's a lot of context that needs to be taken into account. And the answer is why. Not only can we recognize behaviors that are semantically meaningful, can we identify from behavior alone for example, females versus males, plain zebras versus gravy zebras, and why, what makes them different. And so with that, of course, imageomics is not restricted to animals in the wild, but of course also animals in the lab, plants, plant phenotypes from images in both for ecological and agricultural purposes, and going down the resolution to the cellular or the full habitat level. Biology does not stop at organismal level. So, but in all of these, the driving question is, can we extract the traits so that we can connect the phenotype, the collection of traits, to the genotype, the underlying, and so we can understand why and help scientists see what they could not see before. Thank you.
Thank you, Tanya. Thank you very much for this very broad talk with many details. I think we are running a bit behind schedule, but I would take a couple of questions.
Maybe the oral speakers could already, the first oral speaker could set up.
Any question? Okay.
What kind of explainable AI techniques are you guys using in the pipeline, if any, so far?
Okay, I'm repeating the question. So which kind of explainable AI techniques are you using so far?
Great. So our overarching approach is to leverage the biological structure and the biological knowledge, sort of to broadly the knowledge-guided machine learning, but to leverage the biological knowledge to constrain the embedding spaces in various ways. So whether it's in a simple way by using the hierarchy of the taxonomy, or in a more subtle ways of using the phylogeny to guide. There is a new approach that is currently under review that really pushes sort of the overall interpretable AI by leveraging the phylogeny, even sort of as an example. I wish I could talk about it. Watch the space. But the very general answer is, as I said, aligning the embedding space with something that's biologically meaningful by leveraging the biological structure. So whether it's the relationship between the phylogenetic relationship, the genetic, so the mother-child or other genetic relationships, or the evolutionary or direct trait similarity that constrains the, through whichever machine learning approach whichever embedding space you're aiming for, that it has to be constrained by the biology, so that essentially the what's close in the embedding space is close initially also because it's biologically related.
Thanks. Other questions? Okay, thank you again, Tanya. Now we have the orals. So the first oral is from Mohammed Elamot. Are you here? Okay. It's what Tanya was mentioning, discovering novel biological traits from images using phylogeny-guided neural networks. So you can
should I connect or just
share the screen.
Right, see you later.
I have a question. Is there like a way to connect with Tanya outside of a lot of time, or to ask her questions later, or
Good question.
to get, you don't have an answer now.
If you don't have a budget or platform to have offline QA, like no,
Should I unmute myself or?
yeah, please. No, please,
No.
please keep muted. So,
Okay. Do guys online hear me? Okay, cool.
Yes.
Okay, awesome. Cool. Well, thank you everyone for being here. It's an honor to be with you here. And if you see on the slide, this work is the culmination of the collaboration of almost 20 people, including Tanya and my advisor, Anush Karpatne. And this project was part of the Imageomics initiative, which Tanya also talked about in her talk. So me being a computer science researcher, this was a new experience for me to work with so many awesome people from different backgrounds, some biologists, some computer scientists, and it was a great learning experience to me. So without further ado, I'll go ahead and get started. If you look at the title, Discovering Novel Biological Traits from Images Using Phylogeny-Guided Neural Networks. So you can already see there are like 3 probably keywords that we can focus on, which is images, traits, and phylogeny. And maybe some of you are not familiar with all of these terms. So I'm gonna try to take it one step at a time and build it up until to see what is this project really about. Okay, so let's start with images. So first, let's talk about discovering attributes in images. So we know computer vision has been a big thing lately. And in particular, I'm going to focus on autoencoders. So autoencoders take really images from very high dimensional space and crush them down into a latent space that is usually much lower dimensional. Okay, and in many research, this lower dimensional is like capturing attributes or characteristics in the image. Okay, so maybe in this case we have a face image and we crush it down into certain attributes like the smile, the hair color, the eye color, skin color maybe. So what is the use of this? Well, there are 2 uses. First of all, it helps with understanding the image. If you want to take it from an explainability perspective, we can understand the attributes that we as humans care about. But also these attributes can be used then to generate new images that might not exist in the training set, right? Just like GANs do, for example. But you can do that with autoencoders. So that's from a computer vision perspective, okay? So let's put that, let's put a pin on that for a bit. Let's go to the biology side. So if we look at the specimen or species, it has certain traits, and by traits here we mean like usually anatomical traits. For example, the fins, the tail, maybe the eye, the color, the shape of the mouth. So those are all traits, okay? And generally, these traits are the result. I mean, again, I'm not a biologist, so if I say anything biologically blasphemous here, please forgive my sin. I'm willing to be corrected, but generally these traits are the results of evolution over millions of years, and there are also environmental factors, right? But if we take the genetic factors, they're usually encoded as discrete pieces of information, or what we usually call genomes or pieces of DNA, right? Okay, so if we draw an analogy between what's happening here in real life, the species, and here, which is the image, we can see in both cases, we're trying to, there is some piece of information. Here it's the, here it's the vector of attributes. Here it's the genome, right? That captures the information necessary to describe the species or the specimen, right? So that's the analogy here. So the goal of this research is to capture in the image space, we want to capture similar discrete pieces of information, but from the image rather than from in biology, right? We want this to be a sequence of information, just like the genome is a sequence of, you know, DNA. We want a sequence here that describes the image. And there is already some work in the literature that does something similar. So if you know about the VQ variational autoencoders or VQ GANs, instead of doing what regular variational autoencoders do, which is capturing a latent space that has continuous values, in this case, it reconstructs the image using a sequence of codes, and the codes come from a code book. Okay, so there is already some work in the literature that does something similar, which is I want to capture the image in a discrete sequence. Okay, so what is the problem here? What are we trying to do that is different from this? What we're trying to do is these codes do not necessarily align with biological traits. You might be capturing discrete pieces of code, but these pieces of code may or may not align with the traits that a biologist expect. Okay, and we all have seen examples where, yeah, the latent space can learn something about the image, but it might not be something that is meaningful. You all know the dog detection experiment where the computer vision model was actually looking at the background, the snow, to tell a dog from a wolf, right? So it is important not just to reconstruct the image using some embedding space. We want that embedding space to be grounded in the biological knowledge. Okay, so that's really the direction this research takes. Okay, so when we say biological knowledge, what does that really mean here, right? And this is where the other word in the title, phylogeny, comes in. So just like 101 phylogeny here, again, I'm not a biologist, but phylogeny is really just a subset of the tree of life. So, you know, the species are all related to each other, but they evolved over time. Some species are more recently related through a common ancestor than other species. So, for example, okay, you don't see my pointer for some reason. Okay, let's see. Highlighter, you see it here? Nope. Okay, so if you look at these two here, they come from the same genus, right? So they look somewhat similar, right? They look much more similar to each other than this to this, right? And that's because these share a more common ancestor than this and this, okay? And that also means they share more genetic pieces of code as well, okay? So it stands that if we want a discrete embedding space or a discrete sequence to describe the specimen or the species, it should align with this phylogeny, right? If two species are connected through a recent common ancestor, they should share more of that sequence than if they're connected through a far ancestor. Okay. Okay, so this is where our proposed model comes, with just phylo-NN. We call it phylogeny NN, neural network or phylo-NN. Oh, okay. I am signed in here. You just signed it off there. But I think
I tried to sign in.
We can still hear online.
Yeah, my computer is still connected.
Yeah, you can. Oh, yeah, but can you simply connect? Yeah,
Yeah, I'm good. I'm good.
maybe okay.
Okay.
Yeah.
All right, we'll keep going then. All right. So this is the proposed model, which is phylogeny neural networks, or phylo-NN for short. It's grounded in the knowledge-guided machine learning framework, which is generally, in addition to data, we try to add established priors or knowledge in the field. Could be physics, could be biology. It's just any knowledge that is established in the field. It can be used to assist in training the model. And this is generally the knowledge-guided machine learning framework. And in this particular model, the idea is to encode the information, the sequences we talked about. We want to learn that sequence, but that sequence we want to ground it in biology, in the phylogeny, like we said. And this sequence, appropriately, thanks to Tanya and Anuj, we came up with this term, which is imageomics, because image genomics kind of thing. So we want to imagine that the image is made up of this sequence of imageomics, right? As it's like a genetic code for the image itself, rather than for the species in biology. It's just an analogy here, but appropriately so. And this sequence, we can think of it as two subsequences. The first one is a phylogenetic information sequence, just like we said. This is grounded in the phylogeny itself. And the other sequence is a non-phylogenetic genetic information. Because if you take the image of a specimen, there's a lot of traits there that are grounded in biology, but there are other parts of the image that are not related to biology. So it could be the background, maybe the lighting conditions. There are things that not necessarily coming from the biology itself, right? So you still need those that piece of information to reconstruct the image, right? But it's not a biological information necessarily. All right, and the phylo sequence is further subdivided into multiple smaller sequences. So maybe the first sequence aligns with the traits that are necessary, that are common to all species. And then if you add more information, you can add traits that are common to only a subtree, and so on and so forth. So it's an incremental pieces of information, okay? But it's all discrete. All right, so that's really what our model is. I'm just going to go through two results because we're out of time. And then if you want to talk more about this during the poster session, I would be happy to show more. So the first of all, we just wanted to assess how well does this phylo sequence align with the biology actually, right? So we have ground truth here on the right, the distance in the phylogeny and distance in morphology. Distance in morphology means distance in trait space. But we're not going to talk about that now. Happy to talk later. So that's the ground truth on the right. This is like a heat map of distances, right? So you have 38 species here, 38 species here, and this is distances between them. In the ground truth, here is the distances between them in the embedding space. And you can see the phylon N, the heat map is very similar to the ground truth, whereas if we take the BQ GAN, it's not really aligning with that. So we can see that phylon N shows higher correlation with the ground truth. The second one is if we train on some set of species and then we compare to a set of species that the model has not seen during training, and then we see this distance. So we can see that the unseen species have lower distance with their immediate cousins rather than their further cousins, because these two, for example, are post-Natropus. So they share a common ancestor that is recent, whereas these don't. And you can see the distance reflects that in the phylogenetic part. In the non-phylogenetic part, which is this part, you can see the distances are all almost the same, which means the information was properly disentangled. The environmental information or the background information is properly disentangled from the biological traits information. So that's in a nutshell our work. There is more information in the paper. I'm happy to talk about it. It's gonna be published in KDD 23. And my collaborators will continue this work. There is a lot of potential in applying it to other data sets. But that wraps up my presentation, and I'm happy to take questions.
Speak briefly on how do you kind of constrain the latent space between the phylogenetic and non-phylogenetic?
Yeah, the question is, can I describe briefly how I separate the different pieces of the phylogeny in the tree, different pieces of embedding space according to the phylogeny. So if I go briefly back to this figure here, this one, okay? So what I basically say is these species, let's say at this level, share, they get split into 3 or 4 groups here, right? We have common ancestor 1, common ancestor 2, common ancestor 3, common ancestor 4, right? So you can categorize them into 4 groups really. And whatever imageome you have here should be enough to classify them into these four, okay? So if you can just use this part of the embedding space to do that classification, then you have captured the necessary information for this grouping. And then only if you add this part of the information, now you have captured enough information for the next level of grouping. So it's like a multi-level classification problem according to the phylogeny. That's in a nutshell. Oh, there are other regularizers who I'm happy to talk about offline, but this is really the main idea. Thank you. Yes.
And do I mute myself or?
Yes, you're going to use the mic.
Okay, so this should be good. Fantastic. Okay. So all good on Zoom as well, just to make sure. Okay. Well, good morning, everyone. So I will give a brief overview of the work I was doing on how to retrieve camera trap pictures containing specific attributes and using text. So when we're confronted with such camera trap pictures, most common machine learning applications would actually try to localize, detect the species, as well as count the number of individuals. But there's a lot more information that is relevant to ecologists that is contained in this picture. For example, the behavior of the animal. Here we have a giraffe moving away from an acacia tree, one of its primary food source. There is also the forage quality, where we see like a greener grass at the base of the tree and dry grass around it. So how can we retrieve this precise information that cannot always be put into categories with machine learning? So we decided to follow a recent line of work that basically use language vision models such as CLIP that basically do this in what we call an open vocabulary scenario. So these models are being trained with millions of image and caption pairs that result in an embedding space where the information that is represented both by text and images will point towards the same location into the embedding space. So in simpler words, that means that what we want to describe and collect in our images can be described through text. And to find this similarity, we just have to compute the distance between the encoded representation in this embedding space. However, it was shown by Panthesis and colleagues that these models are actually not well adapted to the visual domain of camera trap imagery, and this is because they're mainly trained on images from the internet. So we decided to go in the direction of how can we adapt CLIP to correctly retrieve the images from large camera trap datasets. And we expand on previous works that already did this per species classification and really broaden it towards multi-attributes retrieval. So the perfect data set to work with is actually a snapshot Serengeti one, because it contains attributes not only for, oh, thank you so much. It's actually a snapshot Serengeti one because it contains attributes, labels not only for the species, but also like the behavior of the animal. And so the image processing we do is fairly common in the sense that we use an animal detector called mega detector, and we get bounding box around the animals and those square patches. Then to generate captions, which is needed to create wild CLIP, which is the fine-tuned version of CLIP, we start from the original annotation, so the species, etc., and we select and filter only the labels we were interested in this first study. So we have the species, the age, the behavior of the animal, the time of the day, and the environment that we manually added. Now from these labels, we build structured captions following a fixed template, so a camera trap picture of Earth. And to bring more diversity, and it also have additional properties during training, we also build combinations of these different captions. So for example, in this case, an animal moving in daytime in the grassland, for example. So in the end, we end up with 31 captions describing our patch. And to fine tune clip, so to get wild clip, we actually built 3.8 of such image caption pairs. So we fine tune only the visual encoder part of this clip model. And this is because we want to keep the text encoder frozen, because even though we fine tune with a fixed vocabulary size, we still want to preserve in later applications the open vocabulary capacities of this clip model. And we train it in the same way as clip was trained, in the sense that we maximize the cosine similarity between positive pairs of images and captions, so the zebra and a camera picture at nighttime, and we minimize the cosine similarity with every other pairs. Now, once we have such a model, we can use it for our task of interest, which was image retrieval. So we pass our test images from different cameras through the visual encoder and our test queries through the text encoder. And we compute the cosine similarity of the embedded vectors between every pairs. And we end up with this cosine similarity matrix, where every row corresponds to how our test images are similar to our query of interest. Now, once we have this, we can simply rank the images based from the most similar to the least similar based on their cosine similarity. And because we have the ground truth labels from the snapshot Serengeti annotation, we can evaluate the performance of the model. Now, for the sake of time, I will not go over the quantitative results, and I'm very happy to discuss these at the poster session. So we started with a fine-tuning clip. We got a wild clip, but it needed quite a lot of annotations. I said 3.8 million image caption pairs, which is not always feasible. So how do we go from this base vocabulary to a newer vocabulary for which we might not have that many annotations? So for example, rare species for which we don't have a lot of images, or maybe now we're interested in new behaviors and we don't want to label millions of images. So how can we use this first knowledge from wild clip and go to a fine-tuned version? So this is what we did, and we used a method that was already existing and developed by Gao and colleagues called clip adapter. But it's very simple in the sense that it plugs in an MLP, so a two-layer perceptron at the tip of the visual encoder backbone, weighted by a residual connection, which is helping basically to preserve some of the original embeddings of wild clip. Now we were interested also in exploring a little bit the context awareness properties of our model. So we did this by looking at what were the top 5, for example, images matching a camera trap picture of a giraffe in the grassland. And we see that both clip and wild clip successfully retrieves the giraffe. But now when we move from grassland to woodland, we see that clip sometimes retrieve the same images, while for wild clip, we really have a difference in the environmental domain from grassland to woodland. And the second experiment we did is basically from an image now going in the other direction and looking at what were the captions that are the most similar to this image. And we see that for clip, it's all species-related captions, while for wild clip, we have a combination of the species, but also of the environment of the animal and of the behavior of the animal. So it's basically describing more of this camera trap picture than just the species. And this week, I would like to conclude my talk saying that we had first preliminary results on how to improve on clip for attribute retrieval from camera trap images. And we will follow this work by trying to really go toward a truly open vocabulary scenario with vocabulary that goes beyond what we have in the snapshot Serengeti. And with this, I would like to thank you, everyone. Thank you, the organizers, and as well as my supervisors. And I'm very happy to take any questions you may have. Yes.
You're using a rectangular box. Is it causing problems with giraffes?
It's actually, oh, yes. Oh, I did a mistake. Yes, so the question is that we're using rectangular boxes. Is it causing problem with the giraffe? So this part is actually not really, we use the annotations that were provided on Lila. So I guess we hold here. And these are not square boxes. It's really like rectangular boxes. It's not really causing problem. Okay, now I understand the question. We are actually padding with background so that it keeps the dimension, it preserves the original dimension of the image. So we're not resizing the box to a square, but we're padding with background around it. So, but really all the animals preserve their dimensions. Was this the question?
If you have a giraffe, it's gonna be very small in the image. You will need a lot of padding to keep it right.
Exactly. So then we just have a lot of background next to it. I don't think there's an ideal solution, and I feel it's still better than just resizing to a square. Yes.
I think of an application. So I'm not a biologist, but so the giraffe example from the beginning, you said the giraffe is walking away from the food source. And if you crop the animals, don't you get problems with not having all the information in the image? And did you try with images that had a lot of background and
it's
small animals on it?
Yeah, it's a very good question. So the question was, since we're working with crop images and we are also trying to describe the full scene, is that not let's say a problem to describe this? And I really agree. We have not gone into the direction of, I think, also including the full camera trap, the full picture. And ideally, you don't even consider just the image, but you consider the full sequence. So for example, I said like it's walking away from the food source, but maybe it was eating from it before or not. And yeah, this was really preliminary work to explore what we can do. But indeed, I think ideally you have multiple inputs. One is the crop, and then the full camera trap picture, and then the full sequence. And that way you have really all the elements to describe your scene. Yeah, thank you. I see.
It's okay. Okay, so hi everyone. I am a PhD student at the University of Bristol in the Machine Learning Computer Vision group, and I'm supervised by Thilo Begart and Majid Mehmedi. So today I'll be presenting the PANAF 20K, a large video data set for wild ape detection and behavior analysis. So it's well known and documented that all species within the grey ape family are either endangered or critically endangered as per the IUCN Red List. And therefore we urgently need rapid, scalable and efficient methods to facilitate the regular assessment of grey ape populations, including their behaviors, in order to support conservation efforts. And so at the moment, the development of these methods are largely limited by a lack of data sets, in particular with the computer vision community. And a lot of the animal data sets with regards to behavior are limited by a number of factors. So one, they lack sufficient scale to capture the full breadth of behavioral diversity. They lack appropriate annotation for ecological and anthropological features. And also they're often captured with footage that is not captured by devices which are used in ecological field studies like camera traps and drones. So in response, we present the PANAF 20K dataset. It's a large 20,000 video dataset, which is annotated in two distinct parts. So it has fine-grained annotations, and it also has kind of broader multi-label annotations. The footage that we use was initially gathered by the Max Planck Institute as part of the Pan-African program, which was initially set up to better understand the ecological and evolutionary drivers of behavioral diversity between and within chimp populations. And so, like I said, it includes 20,000 videos. It's gathered from 18 independent research sites across 15 countries in tropical Africa. and it captures apes in a variety of habitats, over 20 of them, including forestry, savanna, and marshland, at over 100 individual camera trap locations, so trail, termite mounds, algae fishing sources, and water sources. And yeah, like I said, it's divided into two distinct parts, and they can be distinguished based on their annotation type. So the first part is the actual PANAF 20K. So 20,000 videos annotated, all with multi-label behavioral annotations. So the annotations were performed by a combination of experts at the Max Planck Institute and also by citizen scientists on the chimpanzee platform, who were targeted specifically with annotating behaviors of ecological and anthropological importance. So here you can see 6 of the parent behavior categories, and there are 18 of those. And then on the inner ring, you can see some examples of the subclasses that we include. And the other thing in terms of the distribution is you can immediately see it's dominated by a few of the parent categories. So you're going to have this long tail distribution, which is always challenging in computer vision. So here, hopefully I can give you an example. So this is example footage with the multi-label behavioral annotation shown on the left, and you can really see that it's rich in behavioral information, and it contains a number of challenges. So in multi-ape scenarios, behaviors are not associated to a particular ape, nor are they temporally located within the video, which makes this particularly challenging as a recognition task. And so we provide first state-of-the-art benchmarks on this. So we train and evaluate convolutional model and a vision transformer. We initialize all models with Kinetics 400 pre-trained weights, and we group the behavior classes into head, middle, and tail segments based on class frequency. And we report performance for each under mean average precision. And you can see, similar to other animal behavior data sets, that the middle and tail segments suffer significantly in terms of performance when compared to the more commonly occurring classes. So the second part of the data set is a much smaller subset, so just 500 videos, but this contains fine-grained annotations. So this is frame-by-frame annotations for full body location, for correct taxonomic species classification, for intra-video ID for tracking, and also for the 9 sort of actions or behavioral actions that you see here. And again, on the right, you can see still frame examples of each of these. And on the left, you can see the distribution of these behaviors. Again, noting the long tail, even with this small subset of behaviors. And again, just to make this kind of clear, it's not super high resolution, but yeah, the bounding box is obviously the full body location. The color of the bounding box is the intra-video ID, and then the text indicates the behavioral action which is being performed. So again, we provide benchmarks for this. So for ape detection, we initialize all models with COCO pre-trained weights, and we can see here that the newer architectures, things like Convex, perform particularly well, especially during validation, although it's superseded in part by the varifocal net and the SWIN transformer at test time across mean average precision and an F1 score. And then on the right, we provide behavioral action recognition benchmarks, which we evaluate with top 1 and average per class accuracy. And we can see here again that the attention-based mechanisms are outperforming the convolutional ones. And we know distinctly that the drop-off in performance in average per class accuracy is because of lagging performance in tail classes. And so to summarize, PANAF 20K is the largest video data set of wild grey apes captured by camera traps. It contains a rich set of annotations for many computer vision tasks, such as detection, tracking, species classification, and behavior recognition. And we provide first state-of-the-art benchmarks for model comparison for the community to build on. So thanks for listening.
Let's investigate like transferability between the apes and other primates, like using this. Is there any potential for that?
So the question is, have I experimented with transferability to other apes? we haven't, unfortunately. We've just been focusing on getting this data set out first. But of course, within the 20,000 videos, there are 4 subspecies of chimpanzee and also some gorilla in there. So we haven't done experiments to determine performance between intraspecies just yet. But yeah, interesting.
Networks you presented running on video or also single shots, like single frames?
On all the experiments here are 16 frame sequences. 16 frame sequences, yeah.
Recently, there was another work with posture and recognition of individuals in chimps and bonobos. Is this data set related to that or it's a separate work? It's from the same institute?
I believe it's a separate work.
Okay.
Are you talking about the dense pose paper?
No, it's about chimps and bonobo behavior studies using posture and identification information. So I was just wondering that if that kind of information, because it's something that maybe I missed, but it's not in this data set, right? Maybe you have.
No, it's
exactly. So that seemed like complementing data set.
Okay.
So just wanted to mention it.
No, if it is part of the same footage, it would obviously be a welcome supplement.
The post-aspiration adding to this part will enhance the behavior of the behavioral analysis. I think.
So we released a paper last year which explicitly explores the use of dense pose estimation in a kind of triple stream network on the smaller portion of the data set, and we show that it does improve behavior classification performance if you explicitly use pose information. Okay.
The original idea.
The other building we had that.
So the deadline since the talk.
Oh, so you were before.
Yeah.
Yeah, I know because it's why you
Also now, just recently started this really amazing project called FathomNet, and sort of as a larger umbrella, Ocean Vision AI. And it's really trying to build up this sort of system and the community and the data so that we as computer vision researchers can actually contribute and sort of help try to understand life under the ocean. And I'm really excited to see what she's gonna be talking about today.
That's very nice of you all. Well, so I'm very excited to be here. This is only my second CVPR ever. And last year I really just exclusively talked about FathomNet. And this time around, I'm gonna try and give you kind of a survey or a sampling of some of the activities our lab is involved in, but also some of the questions that we're really interested in. And I also want to point out, right, we aren't computer vision researchers. We're really practitioners. We like to use what you all as a community create for our use cases. And so while I show you some of these activities, I really want you to think about where might be some ways that the computer vision community could potentially interact with us, because we're sitting on like massive data repositories, for instance. But first, I need to highlight the group. I'm actually very fortunate. There's a lot of us here. I see Joost, but Joost, Lily, Eric, and Giovanna are big members of, Kevin Barnard are big members of the Ocean Vision AI effort, but also FathomNet. So you'll run into them. We're also doing a FathomNet workshop on Wednesday. I'll have a little more information on that. But because of them, we've got all these active projects. A little bit about MBARI, because I always get questions about what is that institution. It's a really, really small research institution. It's based in Monterey Bay. Obviously, it's in our name, which is a really short 1.5-hour drive between like Stanford University and our institution. Where we're located, it's very strategic, actually. You'll see if you look at the bathymetry maps here, we're really, really close to deep water, where within about an hour sail, we'll get to water depths of about 1,000 meters. So from a scientific perspective, as well as an engineering development perspective, that's really important because you can quickly test things, turn things around, make adjustments, and repeat the process. Please come in, and I know there's not a lot of space. Feel free to sit. And what MBARI does, what we're really focused on is the development of tools and techniques that allow us to understand the deep ocean better. We were founded by David Packard, which I think you all have heard that name before. And so that is really our mission and what we're focused on. And at the institution, we use a lot of different robotic systems. So on the left, we have remotely operated vehicles. This is actually Falcor, but we have 3 different remotely operated vehicles. One of them is rated to 4,000 meters. And we also have a lot of autonomous systems. So these are robots, these are drop cameras, but a lot of these systems are rated to 4,000 meters. So access is the game, and we essentially have that, fortunately, at MBARI. But what I also want to point out is this actually describes kind of the evolution of the activities and projects that we've had in the lab. And I will explain that in a little bit, but really I want to focus first on how we've leveraged these remote vehicles to do some really fine scale quantitative imaging of animals in the deep sea, and then how we're now using autonomous systems to really scale our capacity for observations of ocean life. I have to show you all a video of deep sea animals, and I've highlighted my favorites. If you wanted to check out some more, you can go visit MBARI's YouTube channel. It's kind of insane. The top left, and I always have to mention this in any talk I give, is a giant larvation. And I don't know if any of you have heard of this kind of animal, but the animal is that kind of a looks like a larvae or tadpole. It's what's beating its tail, and then it secretes mucus around it, and it lives inside that. It's a really complex filtration structure. We call them snot palaces for fun. We also have calicophora and siphonophores, and this one is Praia dubia. I don't know if we can turn the lights off, but basically those animals, if they were considered an individual, they would be the longest animals in the ocean, not the blue whale. What you see here in the bottom right, those are midwater worms. We think a lot about earthworms and what they do, but the animals in the midwaters, the worms especially, are completely different. In this case, this worm is a Tomopteris. It swims really rapidly. It can actually stop instantly by arresting all of its legs and then reverse itself by just changing the direction of a body wave that it moves along its legs. So from a robotics perspective, they're really, really interesting for bio-inspired design. And then finally, last but not least, the benthic communities that include, corals, sponges, etc. So it's all of these groups that we have ready access to, especially in the Monterey Bay area. And then high level, I just want to explain why we do the weird things that we do in the lab. And the idea is that if we could combine these three things on the left, right, the creation or use of innovative observation platforms, create state-of-the-art instrumentation that we can affix on those sorts of platforms, and then we will then achieve not only access to novel study systems, but then through collaboration, this could be with biologists, mathematicians, physicists, really delve into deeply how these systems function, how they work, and how that might actually inspire other sorts of technologies. So this theme is bio-inspired design. So that is what the group is largely focused on. But here we're spending a lot of time thinking about how do we do all of these things really well, and then engage domain experts to use these systems and answer their particular questions. So to start, I want to show you some imaging systems that we've developed for our remotely operated vehicles. I'm going to highlight a few systems that are already on board these vehicles so you could think about different ways to interact with this kinds of information. So first, there's a science camera. There's, in some cases, multiple cameras that are integrated on these vehicles. There's also collection containers, so they have ability to not only collect animals, but also bring them back to the surface to do all sorts of lab measurements. And then we also have an auxiliary manipulator. So what this allows us to do is, do collections, maybe experiments with systems that we're trying to study while we are deployed in situ. I'm also in this photo, we have a VR camera system. We've actually collected lots of data for this system. So if you're interested in looking at that, I'm happy to provide it. And then three other imaging systems, which I will briefly talk about for this talk, is a stereo camera system that we've used for our machine learning integrated tracking algorithms for these vehicles. We've also developed a laser illumination system called Deep PIV. So it's a laser line scanning, essentially, that allows us to do some observations. And then last but not least, an instrument called IRIS. It's a light field imaging system that gives us like three-dimensional depth data at 60 frames per second. So with all of that in mind, I'm going to quickly go over DPIV and IRIS. And then with my remaining time, I'm going to try and spend a little bit of, time going over some of the autonomous systems and what we're thinking about when we move into the future of ocean exploration. So the way DPIV works, it's actually attached to the manipulator arm on the vehicle in this case. And there is a housing that you can't see that contains, I think, a 2-watt laser that is injected into a fiber. And then there's some optics in that housing there that then creates a sheet of light. We use red light because that actually helps to reduce animal response to it. And so we illuminate a sheet of light in front of the imaging system, which is, I think, HD resolution at 60 frames per second. And the example, I should also add, the field of view can vary because there's a zoom lens. The depth rating is 4,000 meters. And really what I think sets this system apart from other systems that may have been deployed in the ocean before is the fact that our mission duration really is only limited to how long we put the vehicle in the water. Some places that could mean 24-hour operations for collecting this kind of data. Also, what we do is there's a dye and particle injector, and what that allows us to do is inject some sort of scatterers into the water just to get a sense of what sort of interactions we might expect, and then go in with this laser system to do actual quantitative measurements as well. And some highlights, we've been able to use DPIV in a scanning mode, so a lot like MRI or CT scans. Remember, we're just illuminating a sheet of light. So as you move that sheet of light through an animal, a gelatinous animal in this case, or even the mucus house that it resides in, you're getting individual planes of information that you can reconstruct using image segmentation, a lot of approaches that this whole community is very familiar with, and get three-dimensional reconstructions of these systems, not only getting exterior views of what these structures might look like, but then, and I feel like I can say this, unprecedented views of interior structures inside these snot palaces. And what you'll notice is the complexity, right? There are all of these chambers that go off that central chamber. And what's interesting is they all connect in one location near what we call a buccal tube. That is essentially a straw that the animal puts in its mouth. So these are elaborate structures that the animal is able to create by just secreting mucus and beating its tail to blow them up. And from an engineering perspective, like how on earth is an animal like this able to do that? And then do so rather repeatedly. I think we have guesses that they make these mucus structures on the order of maybe once per day. We don't know that because we haven't had those kinds of observations yet. Another example of how we've used the imaging system is to actually measure the movement of suspended ambient particulate and use that motion of the particulate as a proxy for motion of the water around the animal. This process is called particle image velocimetry. It's a very common technique used in mechanical engineering or fluid mechanics labs around the world. But the idea is that now we're able to do these kinds of measurements, and I apologize because this looks much better in a very dark room. But when we take the instrument and we position the instrument on the animal is what's beating its tail there. And then that outline you see around it is the cross section, right, of that mucus structure. And if you look closely or squint, you can see particles. Wow, Kevin, thank you. I might play that again. I think I have time. So not only are we getting the cross section of the animal within its mucus house, we're also getting the displacement of particles that are suspended around the animal over time. And so using this particle image velocimetry or particle tracking velocimetry approach, you can directly measure filtration rates or volumetric flow rates of what this animal is processing when it's beating its tail. And so we've been able to do these measurements. Now remember, these are all in situ. This particular animal is 400 meters deep. It's about 2 centimeters across. The mucus structures can be up to 1 meter across. And we're having I think it's a 6.5 ton vehicle position a 1 millimeter thick laser sheet on an animal that's about 2 centimeters wide. So the fact that we're even getting this data is astonishing, but then also the level of detail that we're getting from just this one imaging system alone is, I was very excited the first time I saw this. So this is at least the DPIV instrument. It's focused on, it's a planar measurement, right? So it's really good at giving us 2D measurements or 2D particle motions. But let's face it, the ocean is a massive 3-dimensional space with a lot of complexity. Animals cannot be, simplified into 2D structures, although physicists would love that. And so what we need are systems that really give us volumetric data or 3-dimensional data that we can then use further. And so we've done a very, very intensive and comprehensive look at all of the mechanisms or approaches that you can take for collecting that kind of 3-dimensional data. And then given those approaches and then our limitations for trying to get an imaging system like this on a deep diving vehicle that's rated to 4,000 meters, we landed on planoptic imaging or light field imaging, which I don't think I need to explain too much to this audience. But the approach is, at least for a single camera light field, you have a micro lens array between the image sensor and the imaging lens. And what that does is effectively subdivide your image into multiple perspectives that you can then use to come up with volumetric data. So the way this will work is this is a raw image collected from the camera system. Each one of these dots corresponds to a single perspective within that micro lens array. And then using this information, you can come up with depth maps, right? Colors indicate distance between the object and the image sensor. And then you can, I think what I love too about this process is given that depth information, you can also create what we call total focus images where everything is in focus. And then finally, you can get three-dimensional reconstructions of the entire, in this case, particle field. And so this is a imaging system that we built, or we started building, I think in 2018. It's called IRIS for remote imaging system. And the way this works, we have, again, using the ROV manipulator, you have the IRIS imaging system. It's a single camera housing or single housing, which is very different from DPIV, which is 3 housings to deploy. And we have, we're using a Raytrix light field camera. That's a company based out of Germany. We have currently a zoom lens, so our imaged volume can vary from 5 by 5 by 2 centimeters cubed up to 12 by 12 by 6. So still relatively on the small scale for some of the animals we like to study, but a really, really good starting point for us. Depth rating is 4,000 meters. And again, we're using red light to try and minimize animal behavior changes. And then another thing I want to highlight is we have this 25 megapixel camera that takes data at 80 frames per second. So we have data rates on the order of 2 gigabytes per second. So it's lots of data that we have to manage. And then very recently, I think this was last August, we were able to deploy this system at a seamount called Davidson Seamount, which is off the coast of California, but a specific site called Octopus Gardens. And there is a location where you can find hundreds of the same species of octopuses that are brooding. But besides brooding, they're also crawling, like you see this particular octopus is doing, and then also free swimming. And so when we went over this 12-hour dive, we were able to collect iris data of all sorts of different octopuses, individuals doing all sorts of different behaviors. And so we're kind of sitting on a big data set we haven't fully processed. But here is a video showing you all how we position our instrument relative to our particular subject, in this case, a crawling octopus. And try to position ourselves relative to the animal so we're getting the kinds of data we need, but also trying to minimize those interactions, right? We really don't want to change the behavior that the animal is eliciting while we're trying to measure it. And so what this is the depth map from the imaging system, IRIS, and we're collecting data. In this case, I think it was at 60 frames per second. So you can see the level of detail we're getting from this image system. And then what we have to do is, markerless tracking. We can't put anything on these animals to make it much more easy to do feature extraction or tracking. And so, unfortunately, Yoast spent a lot of time looking at this footage, selecting different kinds of features like the suckers on the arms, and actually for the first time being able to do, 3-dimensional reconstructions of tentacle movement during crawling in situ of octopuses. And if you look at the literature for like octopus-inspired robots, most of those robotics papers are, and we're talking hundreds of papers, are derived from 2 to 3 in situ studies of octopus behavior. So already the kinds of data we're able to collect here really will, I hope, kind of change and improve our capacity for generating this kind of information of animals in their natural environment. The other thing that we haven't had any time yet to pursue, but is next on the docket, is looking at the outputs for particle and animal fluid interactions. So the idea here is we're getting 3-dimensional particle representations within the scene, and that combined with the surfaces of the animal itself can give us an indicator of like how does the animal movement affect the fluid environment around them as they're swimming or as they're feeding, which is an important metric for understanding their mechanics. And then finally, a fun outcome, but this is where we could really use a lot of help, is taking these light fields and constructing from the depth maps, the 3D reconstructions of the animal surfaces. And the reason why this is really, really important is that we can take these 3D reconstructions and bring them into computational fluid dynamics solvers like immersed boundary methods, for instance, to actually evaluate movement and how like energetics or efficiency changes as you vary some of these mechanics parameters. You can do that within a computational model. You can't do that in situ with real animals. And so what we're trying to do through this approach is to provide essentially labeled data, but validation data for these computational models so that mathematicians and physicists can then run with them and evaluate a variety of metrics around these organisms. So if you have any ideas, I went to a monocular depth estimation session earlier today, and with the ML tracking camera system, the stereo camera system, plus this kind of camera system, we would have essentially a labeled data set for monocular depth estimation, for instance. So things to think about, data we're already collecting for very, very different use cases, but could be useful, I think, to the community here. And then last but not least, and I'm going to blast through this because I want to leave some time for questions. Really what's starting to happen within our group is, while we started on using remotely operated vehicles primarily for all of our work, there is more and more of an interest for us to be doing these kinds of activities or work on autonomous systems for some very, very good reasons. So this is a plot that shows the state of long-term biological observations in the ocean. I think I use this paper in almost every presentation I give now. And the color indicates, in worldwide databases where you can access this data, regions where you have long-term biological observations. So, but it's also constrained to just near the ocean surface. And what the conclusions from this paper was that about 7% of the ocean surface area is covered by long-term biological observations. So only the surface of the ocean, and even that surface, we've only covered about 7% of it. And then the ocean comprises, 97% of the habitable ecosystem on our entire planet. So it's not a surface, it's a massive volume. And then what makes it even more challenging is about less than 1% of it has actually been visually explored, either by a human or by imaging system. So those numbers are awful. On top of that, it's estimated that anywhere from 30 to 60% of life in the ocean has yet to be described, AKA is unknown to science. So think about all of these unknowns, think about all of these gaps. How on earth are we able to achieve filling these gaps if we continue to use really expensive, often difficult to operate systems like the remotely operated vehicle I just showed you? What we need are autonomous systems doing essentially equivalent missions, similar to what we do on remotely operated vehicles, and then running those emissions to scale. Now, if we are successful at doing that through autonomous systems, all of a sudden we're collecting lots and lots of visual data. And I think this room can easily appreciate that. And we did a sampling of how much visual data is already available or exists in the community. So querying the US exploration-based institutions that generate visual data, there's more than 300,000 hours with less than 15% of it being processed, i.e. annotated. And then if you want to fill these gaps, you need to have an exponential increase in data collection. And so that means we're just facing a deluge of visual data. So how on earth are we going to solve that? Well, this is our first attempt. FathomNet is a data set that is really built to be a distributed data source where people from different parts of the world can contribute their image data or their expertise to help us label all of this information. You can check it out on fathomnet.org. Thanks to the fine-grained categorization and visualization workshop, this year is our first year to do a FathomNet competition with Kaggle, which I think the results were just presented today. So this is the first time we've done this, and we're looking forward to continue to do that because, as you can imagine, there's a lot of use cases and challenges with ocean imaging. Another thing I want to highlight is that we are doing a FathomNet mini workshop on Wednesday. So the first 50 people that comes to the Vancouver Aquarium will give you a ticket and also involve you in an activity to help us build the data set even more with animals that are at the Vancouver Aquarium. So please check that out. And then, as Sarah had mentioned, FathomNet was really just the beginning. As we talk to more and more users, people who create visual data, who need to process it for very, very real reasons, like biological monitoring is, as you saw in that paper, almost non-existent. What we've been focusing on is building out Ocean Vision AI with the goal, right, of building data pipelines and architecture for visual data coming in and annotated data coming out by using collaborative interactions between AI and humans. So as part of this approach, we've been funded by the NSF Convergence Accelerator. We're really focused on human AI interfaces that span the gamut between experts and enthusiasts, with creating 3 different software products that achieve this kind of interaction. The first is FathomNet, as I had mentioned, and thanks to the NSF funding, we've been able to really focus on what should be the community interactions to provide motivation or attribution for people to contribute to the database. The one in the middle really is called the portal, and this is where if you have visual data, really it's a digital asset management system that does not exist at a large scale in the ocean science community. And so that is also being built. And then finally, something I'm extremely excited about is a mobile video game that enthusiasts will be able to play and help label a lot of content. And so all of these things together are coming in to try and fill these gaps around human verification, which frankly is more of a challenge, and then hopefully help us really scale our observational capacity using imaging in the ocean. And so some quick highlights. So already we've used FathomNet data in a project called ML tracking. This is where we've trained some supervised models, incorporated them into our vehicle controllers. And here you can see a demonstration of it on our remotely operated vehicle mini ROV. And we're tracking, in this case, a solstice jellyfish, and we're able to ignore or robustly track it as other objects swim by. We've also been able to incorporate this into the same vehicle, but in the case of trying to search for particular objects we want to study. So you'll see in the top left the different modes the vehicle is in based on what the object detector is telling us. And so in this case, we're searching for giant larvation. Surprise, surprise. And then as we come up on the animal, again, this is like 400 meters deep. This is in the wild as it gets. You can see there's some proposals. If the object matches our search class, then you'll see us transition from acquire to track. And so there's so many things we would love to do with this, right? It's one thing to have supervised models running on your vehicle. We really want to look at, open world object detection. There's a whole session today on open world tracking. Like those are those types of approaches we would love to pursue using these systems that we already have in place. So what we're also doing is we're building imaging payloads for autonomous systems. So this is the long range AUV. This is the chitin imaging system. It's a 7-camera payload. So that will give us multi-view perspectives as we're conducting missions. And then the goal, right, is to really piece all of this together into what we're calling supervised autonomy. So if you have a submerged asset like an AUV or an autonomous system, and remember, the ocean is a GPS and communications constrained location. You can't just push data up into the cloud and have it process and then give you information or decisions that you can then make on that data later. So what we actually have to do is we have to create like a data relay. In this case, it's an autonomous surface vehicle that's tracking your submerged asset. And through acoustic comms, you can actually transfer information. You have to make decisions around what that information should be, because again, despite having comms, you're still looking at maybe a couple kilobits of data that can be transferred between these two systems. Then once you get it up to the relay, if you have an image that you really want someone to review, if it's an object that's novel that your object detectors haven't seen before, then that could be sent over satellite connection to a user on shore that then provides some sort of feedback. Is this something that we should be tracking, or is this something we should be avoiding and moving on? So then once you get that decision from a user, that goes back to the vehicle and the process then continues. And so it's this whole ecosystem that we have to create if we're really serious about pushing ocean exploration forward. And the group has already demonstrated bits pretty much all of the bits of this process. It's now a matter of getting this all to work together, but also to do it as effectively as possible. So if you have any ideas or suggestions, I would love to talk to you. And you can imagine we have a lot of things on our plate. And if you are interested, feel free to reach out to me or check out the lab. There's also a lot of different ways you can interact with us. Mbari has an undergrad intern program in the summer. We also have a competitive postdoc fellow program. And so, feel free to take a look at those things if that should interest you. So I'll take a question or two. Anytime.
So fascinating research. We know that the plastics are threatening life on the oceans. Is there a use case where you kind of study this also with this kind of approach?
Actually, yes. We've done, remember I mentioned that DPAV has a dye injector. And so a study that was done with collaborators at the Monterey Bay Aquarium, we showed that giant larvaceans will actually consume microplastics. And because they're so good at filtering particles and food around them, those mucus structures wind up being kind of aggregates of microplastics in the ocean. So we also find microplastics everywhere in the water column. It's not just a sea surface or a seafloor thing. So it's a big challenge. But thank you. Hi.
In addition to all the video sensors you have there, do you have any acoustic?
We do. We have plenty.
Do with the acoustic data?
Well, that's a good question. So we have a number of acoustic payloads. For instance, we have an upward looking sonar that it's called DEMOS, that is constantly collecting data in the Monterey Bay area. I think it's at the Mars node, so about 600 meters depth. And so, that's tracking movement of vertically migrating animals, for instance. But we have payloads on our AUVs as well as our ROVs as well. Kelly Benoit Bird is actually the lead, I guess, scientist at Mbari focused on bioacoustics data. Way in the back.
Can they reproduce that mucus somehow synthetically? Can that be a way to filter microplastics more efficiently?
You're not the only one who's asked me that question. And I don't know it's possible. I mean, I think for me, at least what I'm really interested in from like a mechanics perspective is why do these animals have such complex structures? Is it a passive mechanism for particle selection, either by size or by content? I don't have the answer to that yet.
To the full autonomous systems. Things working against us is that economies of scale.
Yeah.
Quite niche. How do we actually get there economically?
Yeah, there's a data problem, there's an access problem, there's a hardware problem. And really what we've pivoted to during the pandemic, et cetera, is focus on the data problem. That's why, FathomNet now exists. We're focused on that for Ocean Vision AI. But we're starting to see some movement in the low cost hardware space. There's a lot of people trying to build, low cost image sensors that are either drop cameras or can, have float capability so they could change their vertical position in the water column over time for low cost. I think one of the few groups that have managed this really, really well is the physical and chemical oceanography community. If you want to look up the biogeochemical Argo or the Argo float program, this is where you have these autonomous floats that just have a buoyancy engine that change the depth of their observations. And they have a pretty significant coverage, not through the major ocean basins, but also more recently through the Antarctic basin as well. So we essentially need to get to that point. But in order to do that, we really need to think about the algorithms we deploy how do we down sample the data we're collecting so that the useful information gets shared in these, data and GPS constrained environments. Cool. Thank you, everyone.
I am, yes. Just need to plug that in. You can. I need to get my add-on. Okay. It's too heavy. This will never work. No, but it's too heavy. Too heavy. Yeah. Okay.
So with here and also here.
Yeah.
Okay.
Should work. Yes.
Yes.
Works. Oof. Sta quasi. Screen should be. Oof, worked.
Yes, good.
I just need to take this out. Oh. Good.
That means, I have of the environmental computational science laboratory, CVPR, where he studied the Earth from above with machine learning and computer vision, and I think
also from below now. I need to change that description.
in the team, he became assistant professor.
Thank you, Sylvia. Thank you very much. Hello, everybody. How are you doing? Good? Okay. Very difficult to go after you. So I'll do what I can. So thanks for the nice introduction. And today I'm going to talk about something different. I mean, if you have heard me talking before, maybe it has happened. I usually talk about terrestrial problems with drones. And this is a new project that we are running since 1 year after, more or less. And I'm very excited. That's a new thing. So, I mean, indulge me if I say something stupid, but I do what I can not to. So the idea today is not only to show you beautiful pictures of coral reefs, but also to see what we can do to monitor that in a very special ecosystem, which is the Red Sea. So let's try to pass the slides. Yes. So again, we talk about life underwater, which is something new, at least for me, and in particular, so this is very relevant for Sustainable Development Goal 14. And among all things we can talk about life underwater, we'll talk about coral reefs in protection, restoration, and conservation. Right. So why this is important? Again, I might repeat something that has just been said, but the corals are not just pretty. I mean, they are, but they also are very, very important for our planets and our health for a number of reasons. So first of all, it's a source of food, directly or indirectly. So directly because fish the corals, and indirectly because we eat the fish. So since the fish is the main source of protein for many of us, protecting corals, it's a good thing to do to keep that chain untouched. Then we don't want the microplastic pollution. So also it is important regarding the fish. For the biodiversity, well, coral reefs are not very much. They do not cover very much of the oceans, but they host a quarter of marine life. So actually, it's pretty, for me, important that we make them survive, because it's the house of the fishes. And finally, it's not only about fishes, but it's also of the protection that corals give to the coastline, because they break the waves and they help us protecting from erosion. And then they're also beautiful and support tourism. So there is a lot of ecosystem services that corals give to us. And despite all this, well, we have basically lost half of the corals worldwide in 50 years. And okay, there's also problems of plastic pollution that are further exacerbate these problems. So the question that, of course, we are at CV4Animals here, and talking to an audience that it's probably more, which is easier to answer these questions, but I'm pretty sure we all agree that, you know, the future on the left is the one where we want to be, and not forcedly the one on the right. And you will be surprised, I mean, how far these two places are. They're not far at all. These two pictures were taken in a single dive. Okay. So basically, corals are under threat, okay? And they're under threat because of a number of reasons. So first of all, there is the water temperature increase, which is due to a number of factors, and they are quite heavily anthropogenic. And it's not on, and more on top of it, there is also all other kinds of stresses that can be the pollution of the water, or the exaggerated usages of some reefs for tourism. And I don't know if are you divers? Who's a diver in the room? Cool. Okay, half of it. Very good, very good. So when you dive, you don't touch them, right? But if you go to a very touristic place with coral reefs, you can tell me if people respect that very simple rule. Probably not. So all this together makes that, you know, beautiful places like this are already not so easy to find anymore. And if you continue like that, there probably won't be many left. So this is why, with some colleagues at the EPFL and in other institutions, mostly in Europe and in Israel, we've tried to really concentrate on the Red Sea as an ecosystem to study. First of all, of course, there is a practical consideration. It was the closest, but it's not the main reason, obviously. It's because the Red Sea, it's a very unique ecosystem in terms of coral life, because our Israeli colleagues in Eilat have made this discovery, which was quite interesting, is that corals in the Red Sea resist to increasing water temperature much more than their cousins in Australia or wherever you look. So if corals in Australia bleach at 1.5 increase in temperature of water temperature, in the Red Sea, they resist up to 4 or 5 degrees. Which is, I mean, it's a significant extra resistance. So we're basically trying to find why is that, okay? And there is a number of hypotheses we have, of which one of the most promising is now mostly genetic and historical, due to the fact of the the Red Sea is a closed sea that has only one entrance, which is here at the bottom. And back in the ages, the Red Sea was lifted, and so it became a kind of a pond without any inflow of fresh water, which killed basically all life in the Red Sea. Then the land subdued again, and it was again filled by fresh water by the south, and it was recolonized by corals. But which makes that the corals had to migrate up north, and going through this very, very warm area here. And they had to adapt to this warm temperature, which makes that now we find corals that are very resistant to it, even in the north where the water temperature is much lower. So that's our main hypothesis. And we're trying to, confirm this. And basically we're going quite often actually to take samples and map the Red Sea ecosystem. So we have this transnational Red Sea Center of EPFL, which is supported by the Swiss government, where we have we're trying to basically creating this regional monitoring network of the corals together with all the countries that are in the regions, which we train by going there. We make seminar, we train divers that are from the region, and we try to set up a monitoring system where basically it's not always us going there and showing everybody how smart we are, but we're actually training people there and making sure that there is some capacity building, and then everybody is empowered and works together to this type of monitoring actions. And it's working pretty well. So we've been doing that for now a couple of years. So 1 year of substantial work, and there we are. So of course we are at CV4Animals, we are at CVPR. So I will talk less about the diplomacy part of things and how you make different countries on the table for conservation. But I thought of talking a little bit more about our monitoring system, which, as you will see, I mean, it's extremely simple, and contrary to what has been maybe shown a second ago, which is fantastic. But I will explain why in a second. So we started with the idea that, I mean, we're not the first mapping and monitoring corals through imaging, but maybe one thing that this previous systems didn't have that we do have is that they were pretty time consuming. So here we are talking about a system that to map a 28-meter plot times 6 meters was taking more or less 21 hours to create a 3D model of this pretty tiny section of the reef, which was done in a fantastic quality and resolution, but it was difficult to scale that up. And then more recently, there are other systems that are very good when we go to the Bentley classes recognition, but then they're based on setups that are extremely expensive. And this is also something that we didn't want to go to, because first of all, you need a lot of money to build a very expensive system. And secondly, because I mean, our idea was to make monitoring that will be repeated and empowered by people that are on site and they cannot afford a $50,000 setup for a nice hyperspectral camera. So basically, we thought, okay, how can we democratize coral reef monitoring by having something that is scalable so that we can apply at several places with very cheap setups, so with very low entry prices of the system, and keep it simple. Okay, that was our bet. And the system we came up with is something based on some cameras that you can acquire on the market. So you go to your favorite electronics store and you will buy a GoPro camera. That is a little camera like this. We have a system. So the Mark I system was a okay, and I have to say it, like a Barbie thing on plastic, and we were putting our cameras on it. Then Mark II actually was with a metal stick, and now on Mark III we are on several cameras mounted on the same. We're basically, filming the reef while diving. So there is a human operator, but basically the whole setup is less than $1,000. So it's something that people can acquire, and we can also leave to the local communities when we go to map in one place. We leave some cameras, we train people, and then they keep doing the monitoring. All right. So computer vision part. So basically, we have built a model that's starting from an ego motion video, so the video that is taken by the GoPro camera, performs 2 tasks. Okay, one task is the depth estimation, so the estimation of the depth of what we see, and the second one is the semantic classification of every single pixel in each frame of the video, and then we use them concurrently to construct a 3D model. So of course, one of the two tasks is supervised, the one at the bottom. So we have our students. The advantage in a university is that we pay our students. We pay our students. And sometimes our PhD students, hi Valentin, to make the the annotations for us, to creating this very well-performing semantic segmentation models. And the good thing is that again we try to keep it as simple as possible so that we can 3D reconstruct in video playing time. So it means that the model doesn't have to run for a million hours for one video. If the video is 30 seconds, in 30 seconds the 3D model is done. So that's we're almost there. We are 18 frames per second. At 24, we can say we are in plain video time. So very close. And we tested in 3 places already last year, in Israel, in Jordan, and Djibouti. Okay, so there are 2 parts. The first one is the depth estimation. Okay, so in the depth estimation, you need to do depth, you need 2 eyes, and so we do monocular because we have 1 camera. So basically, we play the trick of the learning depth estimation, and which means that we take 1 frame, the following frame, both times we estimate the depth. Then with the homography between the 2 poses that we estimate as well, we project 1 depth estimation into the other, and then we have some errors to backpropagate. Okay, we do it frame A to frame B and frame B to frame A, and then we can have concurrently the prediction of the pose and the prediction of the depth. And the encoder is based on ResNet if you're interested. And and not only we use when we encode for 1 frame, we not only use that specific frame, but we use a concatenation of 7 frames. So the one we are looking at, the 3 before and the 3 after, we concatenate them into a context encoding plus the encoding of each single frame, and then we use them all together for the 3 tasks. Okay, it doesn't change much, honestly, on the results to do this trick or not, but it doesn't add much to the computation either. So why not doing it? Okay, and let's hope that this goes, because Jonathan sent it to me during the night. Yes. So here you can see a little bit the results. So everything that is blue is close by, everything that is red is far away, and this is the result that it is calculated in video time by the algorithm. And here we are looking at the reef in Eilat, Israel, if you want to know exactly where we are. Okay, so this is the top part of the diagram. How am I doing time-wise? Okay. All right, and then this is the second part, which is a semantic segmentation pipeline. And here we use something that is basically off the shelf. So it's a ResNet backbone, and we use a Unix structure, so that at every single pixel we can do the predictions of the different classes that we are looking at. So the annotation looks a little bit like that, and these are the results of the neural network. So you see, we have a number of classes, and not all of them are Bentic classes. You see, there is, for example, a transdecline that you can see in green here. It's not something that we use to reconstruct the reef. But if we don't want to use it to reconstruct the reef, we need to detect it. And the same goes for the fishes, for example. So this is why we have all these strange classes. For example, the human class. So you can see here at Guilhem, our head of operations, who has very gently accepted to pose for us for this frame. And the story is that we are basically detecting human bodies and far away. Everything is far away, that you can see it in blue. It's not forcedly water, but it's something that is very far from the camera. And when we know where these pixels are, when we 3D reconstruct, we can exclude them from the reconstruction, because these are not part of the reef. But so without detecting them, it's difficult to do it. And so this means that we can go from a result that is like this. That is what we were getting at the beginning when we were doing that. We were doing distraction from motion without excluding this human, fish, transdecline pixel, which is a bit of a mess, because you are seeing a lot of parts of these divers in the reef. It's disgusting to think about. And this is the result when you don't do it. So when the person is still a person and not reprojected on the reef. You can still see some of this body even in the last results. And so in the end, all these systems used concurrently, and let's cross finger that this works. Yes, it looks like that. So these are the different parts of the model. So the one on the top right, you have already seen it. Is the depth estimation. Here we have the uncertainty estimation of the classifier, which tells us basically which parts are we sure or not. So so it's a kind of a border detector and faraway objects detector. And here is the semantic segmentation result. That of course we are not playing temporal consistency at all in the semantic segmentation network, mostly because it will add considerably to the forward pass computation time. So in a way, we try to keep it simple, even though the final result is not perfect. But just remember that we have then the 3D location on every one of these points, and then we can do majority votings on multiple frames. So which means that if you're making a mistake in a frame, it's not a big deal. Okay, so this is the type of results that we get at the end. So okay, the transit is pretty narrow because it's one GoPro camera and we dive in very close. But now we have this new system with 6 cameras, so it means that the footprint gets larger and larger because you have multiple cameras. We don't do stereo for the simple reason that the idea is to scale it up then any possible reef. So everybody who raised the hand before, if you have a GoPro, you will work for us, just that you know. And so if you go on holidays and you take a video of a certain reef, I mean, we will make a system where you can upload it, and this will like nourish our dream of a global coral reef map. And the cool thing is that you don't only use the semantic segmentation to filter out pixels on the 3D reconstruction. We also then get the semantic classification of the Bentley classes for free, which basically allows us then to characterize every site with respect of the different composition. Some reefs are more sandy, more rocky. We can estimate how many of the corals are dead, and so on and so forth. I hope I'm on time. Yes, very good. Oof. Okay, so that's it. These are the wonderful persons I work with, and they go on expeditions with me. I mean, I hope I will join an expedition soon. It's planned. It's planned. Yeah, I mean, how to conclude? So I don't have to convince you here that the threats to oceans are real, and also that it's a large share of the biodiversity we have of the planet. So we need to care about the oceans too. And this is important also for us as individuals, because we rely a lot on what the ocean offers us. And there is a lot of data. We have seen it before, maybe a little bit more. And yeah, in this specific project, we are very happy because we're working with marine biologists and ecology persons and fantastic divers in the team. So if you have questions, here I am. That's everything I have for today.
I have a question, Devin.
Please.
It's really cool, and I love the focus on like keeping costs down and like scalability. Do you have a sense for like how good is good enough for these models? Like, what are they going to be used for downstream? Like, what are the important parts to monitor? Understand how to understand genes.
I mean, it's not an easy question. It depends on the goal of each part of the study. So for what we need it, this is good enough, because we need to have a rough estimation of the composition that we want to have on the entirety of the Red Sea and then monitor it over time. So having a shift of a little something due to an error will not change this type of results. But it's also true that we go with colleagues that are geneticists, and they really want to see these genetic reasonings we had about the evolution of the corals of the Red Sea, and for them having the exact location of a part of coral that we have been sampling is very important. But I think the two objectives complement each other, and we could imagine having a system that is more, ad hoc for when we go with the geneticist that will map more precisely, and then having a system like this one that is coarse, but still allows you to cover like larger portions. Yes.
So the colors of objects, coral reefs and underwater change greatly based on the sun conditions, the depth of the water, light attenuation.
Yes.
Did you try to do any color correction or enhancement of the imagery?
Not yet.
No.
We are working on it. But there are some persons in the room that I think that have a lot of experience in that. Hi. We'll talk later. we are looking at it now to color constancy correction and things like that. Yes.
Basically, how do you get the location information for the measurement?
Yeah.
Or this, you know, GPS in the water.
It's I knew I didn't have to come after you. Sorry. All right. So how do we make sure that the transits are at the same place, basically? Okay, yeah. So for the moment, it's pretty rough in the sense that we go with the divers out. We put the line, and we have the beginning and the end of the line measured with the GPS, and so we try to follow a line that is as straight as possible. So as long as we see the line and we run the same transit, we're fine. But if you go back 6 months after, it's not going to be enough. So now we're looking at settings that that have like relays on locations. So, but we are purchasing it, and it will add up to the cost. So again, it will be a trade-off between what we do for our own development and cool research project and what anybody can use. Yeah.
Could you do some sort of landmarking or oral?
We would prefer not to, but yeah, we would prefer not to. But at the moment, we do that for the time we stay in one place. So we put these 2 marks at the beginning and at the end, and then there is a floater, and that floater we measure with the GPS coordinate, and that's for the moment what we do.
Thanks.
Yeah. Emma.
Related to that, actually, is it possible to get inspiration from people who do biologging? And so they measure the depth using the pressure, and that's how they connect the points. So it's like for animals that are tagged with like GPS or whatever.
All right.
But when they go deep.
Okay.
Yeah, pressure sensor and depth sensor, which helps you to get data how deep you're going.
Okay.
Get next point when they come. So they interpolate that data.
Okay. Yeah.
So that data could be realistically available. I don't know resolution of that, but that's used for underwater tracking.
Sounds good.
Yeah.
So the question is if we can use a pressure sensor to improve the quality of location. Pressure and depth. The answer is I want to talk about it. Yeah. Very good. Yeah, I mean, for now, the fact is that we went there and then we realized things and we tried to solve them. That's basically how it happened. Yes, please. It's so how long does it take to reconstruct a dive site? That's the question. It's on the video time. So you run the video, and at the end that you have been watching it, you have it. Plus a little bit because we are 18 frames per second, so it's not exactly video playing time.
So I quite understand how you are estimating the depth of the pipe, which is relatively straight, and you said you will move in a straight line. Moving along the pipe.
Yeah.
2 cameras, they'll always be straight.
You're never. Yeah, but you're never 100% straight when you're in water.
If it is straight, you can't estimate the depth.
I mean, you have the so if you dive in straight, you cannot estimate the depth. That's the question. I mean, you have the. I mean, it's depth through movement. It's monocular depth estimation. I mean, you can do it even if you go straight.
The pipe was straight. You're moving parallel to the pipe, and you said you move straight.
Yeah.
You won't know what depth the pipe is, right?
No, but it's relative depth. I mean, we're not saying this is 22 meters, 0.5. It's relative to the position of the camera.
Because there's no texture on the pipe, and you said you're moving in a straight line, and you see the same image throughout your journey. And
But not because everything is moving towards you. It's not the same image all the time. I mean, the coral landscape changes as you move.
Just talking about the pipe, you said you need to detect the pipe.
Pipe? The line, it's just a string. we're trying to reconstruct everything else. The string is the only thing we don't want to reconstruct, actually. I mean, it still gets in because it's in the images, but we don't need it. We don't need it in the 3D model because it's not part of the reef. So we basically remove it. Yeah.
You said we can detect it to remove it.
Yeah, by the classifier. Okay, now I get the question. Is the classifier that detects it? So we have a class line and we classify it. Yeah. Yeah. Sorry, I didn't get your question in the first place. Yeah. Yes.
So is this data public?
It will be. So is the data public? Yes, it's part of our engagement to make everything public. We're still processing it. Yeah. In principle, yes.
So now that you're on that slide, can you give an example of what classes you segment there on that?
I knew. Yeah. I knew I should not have changed the video this morning because Jonathan prepared a prettier one. But now that I have done it, I will try to explain you a video that I've seen once this morning at 6. So the line is in the bright green. We have the massive corals here. The fishes should appear anytime soon. So we have the fishes there. It's difficult to do that in real time. So this is the sandy bottom. Let's see. So basically, we have the massive coral, the soft corals. We have them when they're alive, dead, and bleached. That's more or less the classes that we have. Yeah. So this is like classified as sand, for example. Yeah. But it's true that this video that I will try to stop now does not correspond to this color code because that was the old video and I just changed it this morning. Sorry about that. Yes.
Then do you have objects that are kind of man-made objects staying in water for a long time so that they are,
yeah,
an ImageNet would not detect them anymore, for instance?
Yeah. So the question is if there are objects that are deteriorated because they have been in water for a long time. Yeah, I mean, the model is fine-tuned with this type of categories. And as you can see, there is a category that I'm looking that's rubble. Yeah, rubble. So we have a category rubble and we learn from it using those frames where we see a lot of, unfortunately, plastic and things like that. So we train the model to recognize them.
So you could actually couple this with a, let's say, robot that will pick rubble eventually.
That would be amazing if we could couple that. So the question is, could we couple that with a robot that will pick up the plastic? That would be super cool. Okay, thank you so much.
Okay, so then we went on to giraffes. I've seen an amazing amount of sea life, and now it's time to introduce to our scene horses and dogs. So it is a great pleasure to introduce Albert Ali Salah from Utrecht University. He has made numerous and really inspiring contributions in the field of human behavior, computational analysis of facial expressions, interactions between parents and children, doctors and patients. And he's also behind the newly established AI and animal welfare lab in Utrecht. And this will be the topic of today's talk. So welcome.
Thank you, Anna. Thank you. Okay. Is it visible also in the Zoom, do you think? Something weird?
Right. What can you stop sharing the
it's interesting.
Restart sharing again?
Yes, I will try to do that. I need to go out of this.
Can you click that stop sharing?
I don't see it here, but yeah. Share.
Oh,
didn't work. No, it doesn't work.
Why?
And show stop share.
Oh,
And now it's showing. Okay. So let me try to do this again, maybe. No,
no,
it's very weird. Okay.
Yeah, maybe
So now you can stop the Zoom sharing.
I could.
And you start the presentation first.
But then I will not be able to.
I think you can. So
Okay.
you can simply task switch. So command plus tab or something. And Zoom.
And then start sharing.
Yeah. It's specifying the second monitor. Fantastic.
Okay, that works. Yes. So the title of the talk is completely a misnomer. It's not about AI and well-being in general. It's mostly about dogs and horses. But I want to talk a little bit about the AI and animal welfare lab that we have just announced a couple of weeks ago in Utrecht University. And then I will talk a little bit about pain estimation work that we did in Utrecht, and then conclude with some challenges that we're facing. So this welfare and AI and animal welfare lab is one of a series of AI labs recently established in our department, and these are interdisciplinary labs. And this one is mostly led by people from the veterinary science faculty. From our department, me and Remco Veldkamp are involved. We're both working on computer vision, multimedia, and multimodal interaction. But we have a bunch of veterinary scientists, but also animal ethologists in the lab. And some of them are pretty big groups with a lot of history of research in these areas. And the aim of the lab is basically to create some animal-centric artificial intelligence-empowered models and applications. It's all very hands-on, very practical, and very much directed towards the goals of the veterinary faculty. There are a couple of thematic focuses. I'm presenting you this because I'm hoping maybe there will be some future collaborations between our lab and with people in this room. We're already discussing with the University of Haifa to start an ITN maybe on educating the people that have both the veterinary and computational skills to work in these areas. And the thematic focus that we are working on more is locomotion and activity detection and recognition, automatic monitoring of welfare, and human-animal interaction. The horse research that kind of got me into this is actually pretty old at Utrecht University. The first veterinary faculty was established in 1762 in France, but very shortly afterwards, in 1821, there was one established in Utrecht, which became part of the university in 1925. So the horse research is quite old at the university. And over time, it has also developed into certain focus areas, one of which being locomotion and lameness detection. So I'll just show you a bunch of the projects that are being conducted under the aegis of the lab now, like the EquiPro equine gait analysis project, which is in collaboration with a startup a spin-off from the university, working with wearable sensors. And one of the things that make wearable sensors suitable for this kind of work is that the sampling rate is pretty high. So this is 1,000 hertz sampling rate sensor, which is something, you can detect subtle patterns that are not visible to a human eye with this kind of motion detectors. And the group has recently also organized the Canine and Equine Locomotion Conference in Utrecht a couple of months ago. Another project that involves computer vision is 3D printing of hooves for horses. These are things that you do not need to nail into the horse's hoof so that there is less risk of laming the horse. And then you can adjust these hooves to the individual needs of the horse at the millimeter scale. The problem is, of course, you have to streamline this so that it can be quickly done by, let's say, a horse owner or a veterinary. And then there are some issues that needs to be solved because you're producing these horses' hooves from plastics, and every time the horse is going around, you're releasing a little bit of plastic into the environment. So we should think about using some degradable materials in this kind of thing. Improving hen welfare through computer vision. if you have a hen coop, you have many of these animals, so it becomes very difficult to tag them with wearable tags. So we need to rely on computer vision to be able to pick up the sick animals, for example, from their behavior. At the moment, the computer vision algorithms do not scale up very well for this task, or they will require a lot of computing power to individually track these animals. So there is research conducted to improve this. Another project, this one is a Horizon project, a European project called A-Wish, and this is for improving the welfare. Well, it's a bit strange when we talk about welfare in the slaughterhouse, but it's an automatic monitoring of the slaughterhouse, the complete pipeline, basically, on the farm, unloading, transporting, and slaughter. This is, so one part of the lab is also working with companion animals, dogs and cats. And this is research by Saskia Arndt, who is an ethologist, and they monitor nocturnal activities, and they do sleep assessment for dogs. And if you have ever worked in sleep assessment, it's a very difficult problem. It's a challenging problem if you want to do it remotely. Even in humans, it's pretty difficult. But they do, they use some wearables plus the video observation, and they create these ethograms, and they try to describe the sleep activity of dogs. Two projects that I'm going to talk a little bit more about. One of them is pain estimation in equine faces, and that includes horses, donkeys, and mules, maybe. And then the second one is dog pain estimation for camera. So this is a video-based analysis project. Just a few days ago, accepted for presentation in the Affective Computing and Intelligent Interaction Conference. It's going to be in the coming months. So for pain estimation in equine faces, our aim is to understand pain indicators. And then there are a number of grimace scales developed for assessing horse pain from the facial expressions. One of these scales was developed at Utrecht University Veterinary Faculty, and another one was developed in Sweden. And we use both of these. We use actually multiple scales, because in this task, we are trying to do this from images, which is actually quite difficult. But we created a database and then annotations, and we implemented a computer vision pipeline. The two scales that we have used, you can see the references, one from Thijs van Loon and the other one from Dalla Costa. These grimace scales actually include a lot of indicators that are based on video. So, for example, some of the indicators require that you listen to the sound the horse is making. For example, the flamen behavior, this snorting or head movements. So they're not possible to estimate from images. What we did was we selected the static features from both of these scales and combined them into a grimace scale, but that is based on images. And so you can see here the 6 areas on the face of the horse that we're looking at: the ears, the orbital tightening, the eyelid, sclera, corners of the mouth, and the nostrils. And each of these things, these locations, are scored from 0, which represents no pain, to 2, which presents severe pain. So the veterinarians, when they scale the pain, they look at these indicators and do this per area. And then you sum this up to kind of get a score for the entire pain. We collected 3 different data sources we used in collecting our data sets. The horses from the Netherlands is images provided by horse owners over all the country. And then some of them have bridles, some of them have not. So there are quite a bit of variation in these images. Then we have a small data set that is horses with clinically induced injuries. That's an clinically, ethically approved procedure for inducing a short amount of pain for a certain duration. And then we have images of donkeys from a donkey sanctuary in the UK. We wanted to see whether the models that we developed for horses can we use them for other equines as well? Can we use them, for example, for donkeys? And here you see some of the landmarking tasks that were implemented in these frontal, tilted, and profile faces. Now, I did my PhD on 3D face recognition, and I worked a lot on 3D facial landmarking, spent a few weeks landmarking the FRGC dataset that came out when I was doing my PhD. Now, facial landmarking in horses is much more difficult because in one single pose, you cannot really capture the entire face. So it's difficult to use the contextual information. And then a single model, if you try to build for all the landmarking tasks, then it will be too complex. So our solution was to build landmarking modules that are specific for poses. So we built actually 3 of them. And then there is less public data for training. I mean, for human faces, what we do is we, download the IMDb, and then you have a lot of human faces with names, and then you can train a neural network for identification. And then you can strip the last layer and use this for transfer learning, because now it has good internal representations for what the face is doing or how it appears. But we don't have this kind of data available for horses or donkeys. So that's basically data scarcity problem. The horse code obviously has a lot of variations. That's also a challenge compared to humans. But when we came to dogs, that was much, much worse. But still, we have different colored horses, and that's still a challenge. And the bridle will create some occlusion problems. Now, in humans, typically when we do facial landmarking, we normalize by interocular distance to quantify the error. So if, for example, if your detected landmark is 10% of the interocular distance away from the ground truth, you say, okay, that's close enough, we accept it as true. And then when it's further away, you say that's incorrect. But in the horse, you rarely have both eyes visible in one image. So we selected the eye nostril distance as a normalization factor, and we basically created systems measuring the error as a percentage of this distance. Now, the pain score distribution in the data set is quite uneven. So you can see the 6 locations that we're looking at, and the light blue is 0 pain. And you can see that we have very few instances with severe pain included in the data set. And that, of course, creates another challenge. One thing we try to do to improve the landmarking is to use data augmentation. And what we did was we took a 3D horse model, and then for a given horse that we have in our database, we thought if we could align it to the 3D horse model, like do lifting to generate a 3D version of it, then we could image this 3D horse from different locations and then generate synthetic images. Now, this is actually inspired by the charades dataset. If you know that, this is a work that does this for human poses. So they use a human pose generator, and then they just paste the human on these different backgrounds, and you have these floating humans in the ether. But it still improves pose estimation greatly. But in our case, it did not work that well because we had only a single and a very simplistic horse model. Now, among the posters today, there was the bite, which is very promising for us. So I have one student waiting for me in Utrecht who is going to start a thesis on applying bite to improve the data augmentation with dogs. So maybe with improved 3D synthesis models, we can solve some of this problem. For this one, we used Levenberg-Marquardt optimizer to kind of align the 2D image, followed by a thin plate spline model to warp the 3D model to the 2D image. And then we used the symmetry to fill the pixels that we can't see in the 2D. For facial landmarking, for those of you who are familiar with human face landmarking, we used the ensemble of regression trees and supervised descent model of Fernando de la Torre in combination. And then we try to focus on the stable landmarks, because when the pose changes, the contours of the face, they will move around the face. So they are not very good in terms of automatic estimation. It's, you know, it's a rather safer bet to focus on the stable landmarks. So that's the whole pipeline. Then we have first of all a pose estimator, and we use a model that was initially trained in Cambridge for sheep pose estimation. Marwa Mahmoud's group. I think some of you already know this, familiar with this work. And then we have pose-informed landmark detection. So for every pose, we have a ensemble of regression trees. Then we use, because we lack the data for end-to-end deep learning, we use a more classical computer vision pipeline using hog features and a support vector machine classifier. Now, if you look at this, so these graphs show you the inter-rater agreements for 3 human annotators for no pain, moderate pain, and severe pain cases, and for the 6 different areas. And you can see here that for moderate and severe pain, the human annotators do not agree with each other very much. So our ground truth annotations for severe pain are not very strong. That's a bit of an issue, obviously. But apart from that, so our F1 scores are roughly changing from 0.5 to 0.8, depending on where, in which part of the face you're focusing on, we're getting decent results. So pose detection from the horse's faces, that works pretty well. 3D-based data augmentation does not work very well, does not give us the boost that we're hoping at the moment, but there is room for improvement. We're going to keep working on this. Variations in head morphology between the different species and different breeds of horses actually make generalization quite difficult. We tried transferring to donkeys. It did not work very well. So for donkeys, you need to basically train new models with ground truth annotated on donkey faces, was our conclusion. And then we had very few samples for the severe pain class. And it's very difficult also for our veterinarian colleagues to collect data with pain. It's the procedures for inducing pain are rarely approved by the ethics committees. And then you need to just look at the animals that come with pain to the clinic, you know, undergoing certain procedures. And then you have some limited data set. The second project that I want to talk about is dog pain. That's a collaboration with a veterinary faculty in Turkey, in Ankara University. And they have collected data from dogs that came to the clinic with with under certain painful situations. So this is naturally occurring pain, not induced pain, but it's also very limited and difficult in that sense. And the aim is to enable video-based pain estimation. And for that, again, the data set is available from the veterinarians. If you contact us, there is an ethical approval procedure that you need to undergo before you can obtain the database, but it's available. For this, we combine several off-the-shelf and in-house tools. And the data set that we have, Ankara University Dog Behavior Dataset, has 61 videos collected from different dogs from a duration of 6 hours and 45 minutes, roughly balanced in terms of pain versus no pain. And then we have orthopedic or neurological pain, but we do not have all types of pain in the dataset. The annotation is done by a veterinary expert, and you can see the distribution statistics. This is how the data looks like. So all the dogs are recorded in this little room. Most of the time, you do not have anybody present. And the dog's behavior, well, you see that the camera is not very ideal for this kind of settings. And this is something we have also seen a lot with doing work with human subjects. The clinicians would collect data for their annotation purposes, and then the illumination conditions will not be ideal for doing computer vision. We use a YOLO models to find the course localization, and then we use HRNet trained on animal pose dataset for a more precise pose estimation of the animal. And then we get these key points, which we interpolate via symmetry and from basically a bunch of simple rules, taking into account detections in the neighboring frames in the video. And then we normalize the pose. Basically, we take the dog to a uniform coordinate system, on which we have now the tracking problem. Of course, is a much more simpler tracking problem compared to the birds that you have shown us in the morning. But it's still challenging for us because we have very sparse data and very sparse annotations. We use two-stream network. We use a simple LSTM based processing the key points, and then the color images, we use a convolutional LSTM to process them. And then we stack these two feature vectors, and we have some attention layers to combine them efficiently. For baseline models, we use a grid search. We look at a GradCam activation to see what is the model basically paying attention to at the end. And then we have an F1 scores ranging from, F1 scores ranging from 55 to 74, but we can improve these with this proposed two-stream model to go up to 77% accuracy. There is room for improvement, obviously. The dog breeds are have a lot of variations, but this is the first work ever on pose-based dog pain estimation. So the next step is perhaps to combine this with the face of the dog and use face indicators of pain. The multimodal cues, for example, the vocalizations, attention to wound mobility, responses to touch, these are all things we may want to add in a multimodal framework. We looked at the limited number of pain expressions, and then we were very careful not to fine-tune the models a lot and not to use strong, deep learning models. Because of the scarcity of data, this is prone to overlearning. So it's very easy to, go up to high accuracies with a very complex model. We didn't want to do that. Okay, the future outlook. Well, in the lab, we want to focus on a bunch of problems ranging from sensing animals, especially pain, activity levels, medical conditions, and sleep monitoring. The applications that we have a lot of commercial partners working with us. So we're looking into training and interaction programs, home care applications, post-surgery assistance, welfare monitoring in different specific contexts. And then we need to look into the ethical aspects of all of these applications. This is my last slide. And I tried to kind of list some of the challenges and opportunities that perhaps you may want to discuss in this environment. There are some new data sets that include a lot of animals. They are sometimes not collected for the purposes of animal well-being assessment, like LION, but they do contain animal images, and they are a big help for creating key pose detection or segmentation models. But large and clinically annotated data sets, these are missing. So we need definitely this kind of data sets. Clinical validation for computer-based systems is necessary. What we notice is that in the clinical domains, in both medicine and in veterinary science, the validation is much more meticulous. Whereas in our domain, in computer vision, we just take a database, we say, okay, here is the ground truth, and then we start reporting accuracies, which is not enough. You need to really understand where the errors are coming from, and you really need to look into the individual cases. And also in psychology, this is a problem If you look at the texts of the old Gestalt psychologists, they would talk about each of their patients in great detail, because those are the insights that you want. We have this tendency to reduce things in our field, and that could be harmful in the long run. For computer-based analysis tools, we need simple, intuitive, and useful interfaces. That means this is also an HCI problem. So we need to work with the HCI people to create interfaces into the tools that we're developing. AI ethics research that mostly focuses on humans. The implications of these technologies for animals have not been discussed at all. I mean, if you look at the AI Act, there are very few implications for animals. And I think as a community, we should start maybe this, and we should press for doing research on AI ethics for animals as well. Regulations should be introduced and guided for healthy development of AI-based animal applications. And then privacy issues needs to be tackled. For example, if you're working with horses, sometimes you deal with race horses, and privacy becomes an issue because there are huge financial implications of, if you show this horse in pain or indications of lameness, these can have consequences. And then we feel that experts should be educated with interdisciplinary skill sets to develop animal well-being applications. And I've been talking to some of you, and what kind of skills would a PhD working in this area need? That's definitely interdisciplinary. There needs to be some animal ethology, some computer science skills, data science skills, but also statistics and other skills. So we need to develop programs for that. This is my email address. If you have any questions or comments, they are most welcome. And thank you very much. Yes, please.
Yeah, you mentioned that there were clinically approved ethical production methods. What would be considered an ethical method of reducing pain?
Yes, that's a good question. Sorry?
Can you repeat the question?
Yes, sure. So the question is, how can you ethically induce pain, right? So, for example, in humans, the cold pressor pain is an example, or plunging the hand into cold water to give you a sharp pain that is essentially harmless is one way. But in these horses, I think they're injecting something in the joints that induces temporary lameness. So I'm not a veterinary scientist, so I find this a justified question, but I think, I know that there are no consequences, like any long-term consequences to the horse's well-being, but the short period in which the horse is experiencing pain is there. So that's a valid question.
Under the hoof, like kind of stone, if it discomforts, then pain.
Yeah. So yeah, I was shocked when I first saw that the iron hoofs are actually nailed into the horse's feet. So you might say, okay, is this ethical really? But it actually protects the horse from the uneven surfaces, and I think the way they do it is they don't hit any nerves, so that it's not painful. But yeah. Yes, please. The which one? We don't use it, but some of the models that we use are trained with data from that. Yeah. We use Stanford dog and ticks, but I think there's again, we manually annotated some of these, but we need more data basically. So we were going to also look at this data augmentation problem. I don't know it. So is it a recent data set or? Okay. Okay, thank you. I will check that out. Animal Kingdom. Yes.
did I understand correctly for the dog pain project to process individual images?
For the dog pain project, we no, the whole video is annotated. So the veterinarians annotated the videos in the case of the dogs. So we do not have frame level annotations of pain. We have video level annotations. Yes.
Video annotations for horses as well?
Not yet. So one of the things is that we're lacking videos from horses with pain. But one of the things we want to consider for the future is also to introduce a rig for recording the horses that are brought to the veterinary clinic with multiple cameras, so that we can also have a 3D reconstruction. So 4D scan of a horse in pain would, I think, be an important contribution, but that requires a special setup, basically.
Yeah, it looks like you really need 3D or which is way don't have a lot of data.
Yes.
Like in the case of the horses, and you need to have a specialized model for each pose of the head when you could factor out the pose.
Yes.
That I think would be beneficial for these cases where you don't have a lot of.
Yeah, Sylvia's comment is that we need 3D models that work well with because of the data scarcity. If we can lift a 2D image into a 3D model properly and then generate multiple images from that, from different perspectives under different illuminations conditions. So this is something, by the way, we also faced with human faces, right? Some of the signals are very subtle. So the noise level is actually sometimes larger than the signal that you're looking for. And that makes the problem very challenging. For example, if you want to detect sarcasm in a smile, right? So very small things, sometimes the timing of it is slightly off, and that makes it into sarcasm. You wait a little bit before answering. So it's also interaction. Sarcastic horses would be. We would go to cats if we want to detect sarcasm. Thank you very much.
Yeah.
Yeah.
Okay, I should be muted or not muted?
Can we click?
Okay. Can I connect? Yes.
Screen sharing?
Yes, I can totally do that. Okay. Should do my video too? I'll do my video too. Share screen here. That, and then have a slideshow. Close this. Go.
Yeah.
Can I go? Oh, I have to be on. All right. Here we go. Okay.
Yeah.
Okay, great.
Can you simply click the stay part?
Yes. Stay participant.
Oh, yes, good.
Okay.
The first oral is from Duke Myers, work automatic honeybee.
Awesome. Hi everyone. My name is Luke. I'm going to be talking today. Let me pull up my notes. I'm going to be talking today about the research that we've been doing towards automating re-identification in behavioral choice assays that we run with honeybees. This work was conducted at the University of Puerto Rico, Rio Piedras. with a collaboration called Deep Pollinator that includes a bee ecologist, an animal behaviorist, and a machine learning specialist. And we're all about developing tools for automated pollinator monitoring. So, automatic honeybee flower patch assays. What are those? Ooh. Okay, great. So, as you can see on the left, we have these two little plexiglass flowers that the bees are trained to come visit, and they will drink nectar from the center well. And these assays are used to assess decision making for the bees. And we want to be able to link behavioral traits that we see in these assays with genetics. And to do that, we have to scale them up massively. So we're working on automating the data collection for those. So our data collection system, as you can see on the right, we're videoing these assays from above as the bees come and visit these flowers. And our future overall pipeline is to collect video in the field, detect and track the bees in the frame, and then be able to link behavior observations that we make to the individual level. And that requires a re-identification system. So that's what I'm going to be presenting on today. So the video, here's a sample on the left. And we performed offline pose estimation to detect the bees and track the bees. And then we used that pose estimation to extract standardized images to use for our system. When the bees come and visit, they get a little drop of paint from researchers, 1 or 2 drops of enamel paint. And these compensated images centered on the waist key point were used to create a data set of 4,000 images spread across 27 identities. So that's 27 real world bees with their unique paint code. And you can see here kind of a spread of what some of those look like. And we also extracted uncompensated images, as you can see in the top right. And those will come in later, but these are both like part of our overall data set. So because we knew that we wanted to automate these behavioral experiments and have a model that reliably could re-ID new identities as we run these experiments over and over, we treated this as a contrastive learning problem, specifically focusing at the open setting as our really target use case, where the model is re-identifying individuals that it hasn't seen before. So we used contrastive learning with triplet loss to learn a feature vector for each individual, and then be able to re-ID between photos by using the nearest neighbor when projected into the feature space. And then we did this evaluation using both the closed setting and the open setting. So the closed setting is where the model was trained on all the possible identities that it might be evaluated on. But then the open setting, a certain subset of the identities are reserved to evaluate how the model can re-ID new individuals. And that's further split into the reference and query set. And so this evaluation was performed using galleries of 10 images. You can see in the top right where the particular images are sampled from in each of the settings. But basically, there's an anchor image that is selected from the test set, and then 1 correct image and 9 distractor images. And these are all embedded into the feature space. And then the nearest neighbor is selected to see if it matches the correct class or has the correct identity. In this particular example, you can see that it was able to correctly re-ID this individual, even though the background changed, the pose changed. And so that's kind of where we're trying to create a robust model. And then this example uses full body crops, but we also calculated the top 1 accuracy for 10,000 of these 10 image galleries for several different use cases. So for full body crops, but also just the abdomen, the thorax, and then the uncompensated images that I had talked about earlier. And so this top one performance is plotted here. Closed setting on the left, open setting on the right. You can see these two orange bars show the closed setting performance at top one re-identification for the thorax and full body crops at 0.99. So this shows that we have excellent re-identification in the closed setting using these paint code markers when the model has been trained on all the IDs that it might see. So you could consider this like a more typical classification problem. But we also see surprisingly good performance in just the abdomen crop. And that's really cool because it shows us that in the future, we could potentially use markerless re-identification. I mean, that's definitely like a direction we want to move towards. In the open setting, the thorax still performs the best of all the crops. But you can also see that it performs significantly different than the abdomen and the full body unaligned. And that shows us that in the open setting, the addition of the paint code and the alignment of the images significantly increases the ability for the model to perform correct re-identification. And that makes sense. It's easy to pick out a bee when it has a bright red dot on its back. But it's a really interesting way to kind of bridge the gap between traditional modeling, traditional marking with like tags, April tags, that can be really intensive and hard to do. And then more lightweight methods or even markerless re-identification. So these paint codes, we're presenting a new data set with thorax paint codes for honeybee re-identification. Using contrastive learning, we showed excellent performance in the closed setting. And our next steps are to gather more data to improve generalization in the open setting and also just to further evaluate the performance of this approach. On the right, you can see juvenile bees from days 1 to day 3 can't fly and they can't sting. So you can just grab them by the handful and paint as many as you can grab. So that's what we're working on this summer is getting more paint codes to use and then integrating it into our future real-time pipeline that can be deployed in the field to create these systems for automated pollinator behavior analysis at the individual level to test all sorts of things for the behavioral scientists that we work with. Thanks.
Background and like fake flowers.
Yeah. So the question, totally. So the question is, will we change the background? So we've looked at a lot of data augmentation techniques. These images, I'll go to the data set picture. They're actually 200 by 150 pixels. So pretty small as it goes. And you get a lot of bleed from the background. So even when we do background subtraction, it still really affects the like color makeup of the image. So like the blue and the white are kind of strange, but they're like neutral to the bee. Like the bee doesn't have a preference for them. So for now we're doing this, but when we like the circle image I showed at the end, they're doing that with a bunch of different backgrounds to test how that affects the paint code reading. Does that answer your question?
Did you try or have a look at existing markerless methods like T-Rex or something, which is showing hundreds of fishes identification?
Yeah, so we've done some work with markerless identification, particularly when it comes to the abdomens, because the abdomens do contain features that could be used. But for this particular data set, we haven't looked into it just because of the paint codes, and they need to be monitored by researchers anyways, because they capture them at a certain point. So like full complete markerless is definitely like a future step, but not as relevant as it may seem for this current system. Also, the re-identification using markerless and semi-supervised methods that performed really well for us required a lot more images and a lot higher resolution images. So that's why like we've been working on this approach currently.
Just a small follow-up. So are the color markings also have to be unique? So you have to generate some combinations for people?
So we've been looking at that. So yellow, we have like several different bees that just got a yellow dot. And you can, like a human looking at it, can use the shape of the dot to distinguish it. And while like our model can do that as well, it takes a lot more potential input to get like all the possible shapes. So when we agglomerate all the classes into just all yellow as one class, we actually see really good performance in the open setting. So that's one of our other next steps, is like refining our paint code like choices to really match and be more distinguishable in the feature space.
So like, I was just imagining like a big B column. So right now you have 27 identities. So like for future work, kind of what kind of number are you trying to reach for IDs? So like, is 27 enough, or like you really need 500?
We're gonna need a lot. So like this work is sponsored by the USDA, because this automated pollinator like monitoring can eventually be used to like maybe select for traits that we're interested in, or look at like large scale genetic monitoring. So when you think of a hive, social insects, the whole hive has the same genotype. So like we eventually want to get to a level where we can set out an experiment and then have like a whole hive go through and just like run it over a course of days. So that's like our very far in the future. But we have some promising results on real-time detection and tracking. And so we're coming towards a real-time prototype pipeline. That's pretty exciting. And it's just integrating that into a field system. Yeah.
But the data set, does the data set be generating in the same days or cover a large period?
It's
actually working, or they are isolated?
So it's across 4 different days in the summer. 4 different days, and they're actually like several weeks apart. And then it's like several videos per day. Any last questions? Yeah.
Question about your answer. You said you wanted to focus with maybe the color and more colors, but you should just show that actually abdomen, just using the abdomen, you are able to do classification. I want to just focus on that, and like that you sort of make sure that you can cover so many individuals, and then you can cover eventually 1,000 individuals just using the abdomen.
So what we see right now is that the like lightweight marking methods perform best compared to the abdomen in the open setting. And that's our eventual target case, is to have a model that we don't need to retrain each time we move to a new hive to do this type of evaluation. And so that's kind of our target, and why we're working on more lightweight tagging methods rather than complete investing time in markerless. Thank you guys. Do I have to stop sharing? Oh yeah. Me.
All right, okay. Thank you everyone for staying back for the last talk of the day. So my name is Anuj Karpatte. I'm an associate professor in the Department of Computer Science at Virginia Tech. And my student, Meda Sani, who's the lead author on this work, could not travel. So I'm presenting for her. She's also online, so she can correct me if I make any mistake today. This work is in collaboration with Dr. Bahari Beckham's group from the Department of Mechanical Engineering. This is the Micron base group at Virginia Tech. And what we are doing in this work is very much related to computer vision for animals, but it's sort of different from the other types of animals that we have seen in this workshop. These are microscopic animals, bacteria, that are moving in highly dense backgrounds, hydrogel backgrounds. So the texture The textured background that you see here is the hydrogel. And these objects that are tumbling around, for example, over here, sometimes they appear very prominently. They are the bacteria that we want to track. Why do we want to do this? Well, studying and understanding the mobility patterns of bacteria is useful in a number of ecological and biomedical applications that involve bacteria motion. For example, in targeted immunotherapy, we are interested in loading these bacteria with drugs and seeing if they can tear through tumor cell matrices. And we want to understand what changes in the background, what changes in the pH or other conditions can change their mobility patterns. So if you look at what has been done in this area so far, there are two basic types of works that have been done. The first is where we would do fluorescent tagging of the bacteria so that they are nicely separable from the background that appears black. And this is, of course, easier to perform, but it has its own challenges. First, fluorescent tagging alters the physiology of the bacteria. And second, this cannot be done at the desired frame rates at which we want to study bacteria motion. There is also work that work with bright field images without using fluorescent tagging, but they have all been using liquid medium because it's easy to separate bacteria from the background. But liquid medium is not an accurate representation of the environments that bacteria observe or encounter in natural habitats. For example, inside our human body, which is mostly composed of extracellular matrices of cellulose and collagen and other such matrices. So in this work, we want to study and track bacteria movements in dense hydrogel backgrounds, for example, in agar or in collagen. And you can see that over here, the background is much more dense and it has a lot of texture features. So why is this problem challenging? The first challenge that we encounter here is how difficult it is to separate the background from the objects. Here is an example in agar medium. Right? We have two patches that we have zoomed in, and you can see that for this particular yellow patch where you see the bacteria, you have a nice signature that the bacteria is showing up as a prototype, which seems similar to the feature that you would observe in a background patch as well. And this problem is further exacerbated when you go to collagen, which is an even denser background, right? Here, the bacteria does not even have a typical signature, a typical prototype, and it's very much similar to any other background patch. This is strikingly different from many other conventional applications of computer vision. For example, when you're tracking pedestrians and cars, where the background is easier to distinguish from the objects. The second challenge is that we are recording these movements using 2D microscopy, whereas these objects live in a 3D world, and bacteria can go in and out of plane, right, in a translucent background medium. So what can happen is that a bacteria that was showing up prominently, right, perhaps like a cylinder or like a circle, may become blurred eventually in different frames, and it may again show up sharply in a different frame. This is what we are categorizing as easy, hard, and very hard. And so for every bacteria, we are annotating the bacteria into these classes based on the sharpness of the image or the sharpness of the bacteria. So this is another challenge, which is because of the imaging modality we have. The third challenge is the erratic behavior of bacteria motion. So these are generally modeled as random walk, and this is not as well behaved as the motion of pedestrians and cars. This makes it difficult to extract motion-based features to be used for tracking them. So to address these challenges, we have developed a very simple framework, a proof of concept framework, which we are referring to as MMET. And there are 4 modules to this framework. The first module is trying to augment the features that help in detecting the bacteria. So we are looking at motion-based features for every frame. For example, we are doing consecutive optical flow, and we are looking at the difference from the median background as additional features. Once we have these additional features, we are sending these inputs into 3 different models that are capturing 3 different types of bacteria modalities. We have the easy, medium, hard, defined according to the blurriness or out of focus feature of the different bacteria. We are using simple retina net models for each of the 3 categories. We're combining their predictions, tuning out false positives using a number of post-processing steps. And finally, we are applying the sort interpolation algorithm to track them. So that's a very simple approach. And in our results, we can see that even this very simple approach is able to achieve good performance in terms of precision and recall. Especially on the agar medium, we are able to achieve a performance of 0.82, 0.94 after tracking. And this plot essentially shows how the precision and recall changes with every step in our pipeline. For collagen, our recall is good, but our precision is still 0.51, and that's something that we are working on. Collagen is a difficult background to work with. So here is just 2 videos to show the scale of complexity that we are working with. On the left, you see agar, and the yellow shows our predictions, red shows ground truth, and you can see that it's doing a reasonable job. But if you go to collagen, right, you see these yellow false positives, right over here, which are very similar to some of the false negatives if they show up somewhere. For example, over here, it's the amount of visual information that is contained here is occluded with a lot of noise. So this is a work in progress. We are looking at other approaches, building upon this simple first cut pipeline, looking at deep learning based tracking methods instead of sort. And finally, we are using all of the work that we have done in tracking to go for forecasting or predicting the trajectory of bacteria for different strains with different genetic modifications and so on. With that, I'll like to please. Which figure do you want to?
So on the left.
Right. Yeah. The white. Right. So the question is, what do we see here as the white circles around the bacteria, right? And if you look closely, sometimes you have some really large white circles. If the contrast is showing up well, those are actually diffusion that is happening. And I'm not an expert in how these images are taken, but this is happening because the bacteria is moving in and out of plane. So when you have a very sharp-looking bacteria, maybe in some of the frames you would see like a cylinder type of object or an ellipsoid type of an object. That is when the bacteria is in the right frame. But if it goes in and out of focus, you have these halo effects around it.
And about the data, I mean, you made a pretty good job at showing us how hard it is. But then I feel it's so hard that can you really get investors to ground truth?
Yeah, that's a very good question. So can we even rely on these ground truth annotations? Is there sufficient information in the data that the model can use? Well, that's a question that we are exploring. Our human annotator, which are PhD students working in Dr. Beckham's lab, they're able to identify these bacteria, and they have the expertise. They have the training to identify them. We are trying to do some experiments where we can check with multiple students if they are able to find the same bacteria or not. But certainly there is some information that they are picking up, and we would like to see if a machine learning model can do the same job.
Let's do different kind of video acquisition, like light field LSR here, and light sheet by process to get like volume for this bacteria.
Right. That's a very good question. So I'm not an expert on the imaging, but I can certainly follow up on those directions. We are looking at some directions on changing the z-axis, but it's hard to find the right z-axis that works best for all the bacteria, because these bacteria are not synchronized, right? They are moving in all possible directions. And if we could capture all the z directions at one time, that would be great. As far as I understand, that's the limitation of the imaging that we are doing at the high frame rates that we desire. But I'm certainly happy to learn more about other imaging techniques. Thank you.
Yeah, the end of the
So, Kostas and Albert, I welcome you to the panel.
I think you can discuss with them.
And also joining now.
Check the
Yes, probably.
Or put something in front of it, because otherwise.
Yeah,
Do we have
let's do it.
or
We
projector?
don't,
after?
yeah, we don't need it. So we are. Thank you. Okay, have a seat. No, please go ahead.
From far away.
From far away.
Do we need the mic, or you think it's fine? We don't have enough.
Granted, slightly longer, but it's coming up, yeah.
Coming up. Nice to meet you.
Nice to meet you.
Yeah, share for you.
That's good.
Okay, hi everyone. So welcome to this summary panel of today. And I would like to welcome again Kostas Helges. Thank you. Also our organizer, Albert, Danny, and also joining us, Grant from the University Lab of Ornithology. Thank you for coming. We know it's been tricky for you. So this is the third edition of the workshop, and we have seen today so many people. I think it makes it a timely moment to reflect on where are we going as a scientific community. So I would like to open a discussion and ask some questions for our panelists, but I also welcome your participation. So I would like to ask first question: what are the current challenges that CV4Animals as a scientific community is dealing with? And what is the future vision for this? What do you think we should do as a community? And where do you see this community in 10 or 20 years from now? I know it's a bit difficult. Yeah. I think that Albert, in his final slide, made some comments which resonate with this question. So maybe we can start, Albert, with your vision.
Yeah, I think the big transformation is that the tools that we're developing are now easily turned into actual applications, which was not the case, let's say, 5-10 years ago. And now that in veterinary sciences, for example, people recognize the need to deal with more modern tools of observation. And practically, observation has been a bottleneck for them for many years. It takes a lot of effort. Annotation takes a lot of effort. This has been the same in psychology, obviously, but also in some of the veterinary science problems. So they look for opportunities to extend their toolboxes, which I think is a great idea. So that's what I think is at least for us in the field is going towards. And then we have a lot of experience working with humans, doing a lot of different tasks to understand human behavior at different scales. I mean, we look at individual scales, but we also look at interactions, so dyads or small group interactions. And then we also do computational social science, where we look at thousands and millions of people at the same time. And we have developed pattern recognition-based tools to address each of these scales. Now, I'm guessing that some of these tools are adaptable to problems of the animal kingdom as well.
Thank you. Any comments? Okay, Grant, would you like to go next?
Sure. So I haven't been here today. I've been in another workshop. So just a quick overview of who I am. I'm a researcher at the Lab of Ornithology at Cornell, and so I helped build the Merlin app, if any of you guys have used that, as well as I built the iNaturalist computer vision systems and Seek. So my experience is mainly working with citizen scientists. So yeah, I mean, you asked about kind of challenges, opportunities, where we're going. I think right now, like the biggest challenges I'm thinking about is how do you take these models that have been ultimately trained by citizen scientists and give them to scientists and build up trust in those scientists that the models can do what they originally were designed for. So what I see at the Lab of O is, we take sound ID, we want to apply it to thousands of hours or even more of audio. There's no way we can go through and ground truth the results. So how do you do this kind of active evaluation? in order to give the scientists some kind of reasonable expectations on what's working well and what's not working well, so they can draw kind of accurate conclusions. Because I do think like these models just kind of used in an inappropriate way can lead to incorrect conclusions being drawn from data. And so it's kind of our responsibility to make sure that we not only give them the model, but the tools to figure out how well these things are working on brand new data. So yeah, that's kind of like where a lot of my kind of current thinking is going, is tackling those challenges. And I think the future is just super exciting. I mean, I think all the tools are just going to be getting better. The speed at which we can analyze and collect the data is going to get better. And so it kind of just reinforces this need of that ability to gain trust in all of this. And so, in 15, 20 years, I'm hoping that not only do we have kind of like well annotated data sets that can solve practical real world problems, but we've also kind of gained the support of not just the computer vision community, but ecologists and biologists from other communities are actually trusting and using our tools. So that's.
So you think citizen science is key in this vision?
I do. I think that they've proven their ability to help us annotate data. Of course, there's noise in there and there's bias in there, of course. But I think we've sort of chipped away at the problems of handling those biases. And so I think that is at least one way forward. Doesn't have to be the only way, but yeah.
Okay. Thank you. Kostas, did you want to comment?
Yeah, I would also mention the word trust. I think talking first, it is a great experience for computer science students and engineers to talk to real scientists like biologists, ornithologists, because real scientists go through a lot of pain in order to produce data, which computer scientists have not experienced. And they have not experienced also that get the result might take 3 years, get just one journal paper. So in this sense, I think they regard it as naive. So I think I'm senior enough to say that our results were not good enough. The reason I say it's not good because really they're not usable, they're not automated by biologists. The way they're usable still is that because somebody already annotated what is true, they can use only the good stuff. And still, in terms of geometry, this kind of annotation, they are very helpful. But and they do observations for them. For example, for a physicist, they build an experience with an instrument. This is a very painful process, right? An extremely painful process, which computer scientists are not used to. This is what this is a topic also in the third. I will talk again in the third sentence. But 80%, probably 90%, one of the best percentages I've ever seen today, still actually something that still has for a biologist or a physicist.
Thank you. Annie, can we get your perspective?
I have a lot to add to this. As a not a computer vision person who likes to use the algorithms that the community creates. In my mind, I mean, yes, it means a lot that these tools become easier and easier to deploy and use. But as we were building Ocean Vision AI, we did like very, very intense surveys and discussions like around user-centered design. So like, what were the real pain points that other marine scientists or people who are generating this visual data, like what were they dealing with? And if you talk to this community, it's not just the marine scientists, it's also like contractors that have to collect this visual data to evaluate whether some offshore activity could or could not happen. So like environmental impact reports. These are groups that just don't have a lot of available resources. So you just don't have enough people in your group with the expertise or the time to even spin up these much easier and simpler tools. That yes, it's simple for this community. It's not very simple for other communities. And so I think at least in my mind, this is something that I agonize over a lot, like with AdamNet and some other activities. It's like, how do you actually bring the experts together, right? How do you aggregate expert knowledge and have crosstalk between, you know, these groups or these organizations? Like, I would highly recommend, if you all are looking for use cases, to go either to the Ocean Sciences meeting, which is every other year, or the Society of Integrative and Comparative Biology meeting, which is every year. And these are groups that do phenomenal work in terms of data set generation, collection, instrument development. They know what the relevant use cases are, right? And just having that crosstalk between these two communities could, you know, really open up lots of opportunities. So yes, I agree that the citizen science stuff, the stuff you've been doing is just so inspirational, which is why we're trying to make a video game. But like, I think it's the connecting these expert user groups will be phenomenal if we can solve that problem.
Thank you. Any comments from the audience?
So I've been as a computer scientist working with biologists for the last 5 years. And one of the things that I observe most is that this crosstalk has one issue that most of the biologists, let's say, are still, I mean, when we say using machine learning, there is a very small group. A lot of them have actually problems which are 2D tracking, and it's sufficient to study what they want to study. But they say during the crosstalk, major problem they have is when you talk to a computer scientist, he or she, they're looking to publish in CVPR and ICCV, and they don't find those problems interesting enough because they are like, well, I can't get it published in a venue that I want to publish. So, sorry. And that's the general case. Or if the methods are developed, then they are not to a point where a biologist would be super easily using it. There's only one, I think one of the tools to be mentioned, something like Deep Lab Cut or maybe Mega Detector, which is reaching that level. But most of the tools that are even working on these problems are left at a point where biologists have to figure out so much that they end up doing manual work. So where do you think this connection builds? Like how to do the problems, which then somebody doesn't have to worry about publication, and how does it work?
Don't stop.
Okay, so this is actually a topic I think that came up a little bit during the Visipedia workshop at Caltech a couple months ago. And I mean, if you think about like kind of you bring back to mana, but then there's all these other challenges. Like, what do you do with your data? How do you host it? How do you organize it? Like, and so if you can help solve that problem, right? What that does is fix at least a data accessibility problem, right? So that's, I think, a big barrier between the two groups. Why we focus on FathomNet so much, because it's like, hey, if we can't get labeled data to you all, then you won't be aware of all the variety of use cases that computer vision could have in the ocean domain. So I think that's part of the reason why we focused on data infrastructure. And I think this is what I love to see, like with Visipedia or other groups, is like think about creation of data infrastructure that allow it to be a platform that's ripe for trying a whole bunch of experiments, right? It's not, so it doesn't become, you know, like an impenetrable like chasm that you have to cross on both sides. You're kind of creating something in the middle where, you know, there might be a science use case and they have a particular outcome that they want to see. And then within that same platform, you're then able to plug in all sorts of different algorithms or approaches like assisted learning or whatever to then solve it. So what happens maybe is that you get your data output, the actual need for the scientist or the researcher. And then for the computer scientist, you know, maybe some of the experiments that you need to run to then publish in the CVPR. I think there's so many different ways that you can approach that, but I feel like something like that would be very, very helpful. And already there was a report that came out maybe last week, the Committee for AI, where they were suggesting doing something like this, building kind of this infrastructure that allows people to get access to these tools, but also an aggregator for ideas where, you know, the community can try all sorts of experiments. I'm not saying that's the solution, but I feel like that is one way to bridge the gap. But, you know, it is tough, right? Because I think some of us work on very, very different timescales. If I can get a Science or Nature paper every 2 or 3 years, that's fantastic. I mean, that is really fantastic. I mean, yeah, there is that discrepancy between like how we're awarded, but I think if you can create structures to get us to join somewhere in the middle, I think that might be.
So a comment there?
I have a follow-up question, I guess, to that. Could you talk, so I guess I've thought about a lot of, when coming from similar places, so we thought about similar things, I guess. Can you talk more about, like, even what you were talking about sounded really nice about, like, building these platforms, I guess, to create these kind of integrations. My understanding is that to do that in kind of like a sustainable way, even that takes like at least a handful of software developers and people like that who just are going to be not just separate potentially to sort of academic researchers.
Yeah.
And can you talk about, I guess, one, if that's the case in your experience, and then also if it is, how that works in terms of just kind of funding and I mean, it feels like without being careful so often, you know, you can have these things that kind of last the funding cycle and then disappear. And so just how people think about having that additional work that is
Yeah, no, I mean, I think just following up, I think like there's like always decisions and trade-offs to be made, and you can be strategic in those partnerships that you strike with biologists and ecologists. I know Sarah and I have had great success with just finding folks who have those resources to hopefully get the databases running and managed for longer than just a CVPR cycle or an ICC cycle. So yeah, so our collaborations with the Lab of Ornithology and with iNaturalist and then Sarah with a bunch of camera trapping work has been like, you know, making like kind of being reasonable about the expectations on like what are our timelines for our conferences that we'd like to publish research at, and then what are their goals. And so it's some of this is just like kind of going back to like people communication type things of just setting expectations appropriately, finding those collaborations. You know, like there's a lot of people out there with problems to work on, but some of them are in a better spot to actually sustainably work on those problems, and you know, make sure that data is going to be around and that there's folks that can do the annotations for you. Yeah, and so you know, unfortunately, we can't work on everybody's problems all the time, so we do have to be strategic about which ones we approach. And then, you know, take advantage of those opportunities to then share the results with folks who don't have those resources, right? Getting those pre-trained models out there, getting those data sets out there to give them a leg up when they have the resources to take those steps. But yeah, I think it's just being kind of realistic with your time and resources and being strategic about those collaborations you set up. So.
Yeah, I wish Tanya was here because I feel like she would be able to speak a lot to sustainability. I mean, I can talk to our goals with Ocean Vision AI, but you know, we're essentially halfway through, or more than halfway through a 3-year-long design, development, and launch process. So it's very, very crazy. Have you heard of the NSF Convergence Accelerator? It's a new program that's out of the TIP directorate at the National Science Foundation. TIP is Technology, Innovation, and Partnerships. It's very, very different from other NSF programs where, you know, instead of just running a research program or, you know, trying to answer a particular research question, the outcome of this funding call is an actual product. It could be, you know, a software tool, it could be a website, it could be all sorts of things, but it has to address a relatively big societal challenge in the United States. And so that was actually something we proposed working on, was trying to solve this data infrastructure problem that a lot of the ocean science community faces. But the main driver for it is how we understand the growth of the blue economy. So there's a massive growth in wanting to, you know, do human-based activities in the ocean to exploit certain aspects of it. And the way that is currently being done now, it's being done without a lot of information and data. But not only do you have this need, you also have a potential source of funding. So what we basically pitched is this approach where through Ocean Vision AI, that's the FathomNet portal and a game, it's solving the data infrastructure challenge that a lot of people have. Could be based on subscription models, depending on who you are or how much data you have. But parts of it, like FathomNet and the game Fathomverse, are completely open and public. So through those other mechanisms, you can create engagement, re-engagement, perhaps hooks for philanthropy. Like iNaturalist, I believe, is funded like 50% with philanthropy, which I think is fascinating. And then balancing that philanthropy with, some of the, like a subscription, let's say, is what we're hoping to do. But I can't say it's going to work yet because we would officially launch the whole program next July, essentially. But so it's trying to figure out ways to, similar to your own like financial portfolios, come up with something that's, you've got options besides just one in particular.
I have a comment on the availability of data because so in my group, we work on computer analysis of human behavior at different scales. And the largest scales is that we look at migration behaviors and refugees. We work with mobile phone data. And then we realize that some of the technologies that are based on the data can be double-edged swords. So they can be used to help the refugees or also harm the refugees. One example is you have the application to find from the satellite the boats that are crossing the Mediterranean for helping these boats. But then it's also used for pushbacks by the countries. Now, imagine that you have a lot of ocean data available. And let's say I'm a big company looking for whales to hunt or do fishing in massive scale. And I look at some indicator like that, machine learning is very powerful. Maybe I can use some of the indicators, reduction in plankton concentration, to find large amounts of fish in an area, and then I go and fish them. So the risks are very difficult to assess, especially if you're a computer scientist working on a very specific computer vision or pattern recognition related problem. If you do not understand the social implications or implications of the technologies that you're developing. I mean, I'm imagining that the person who develops a recommender system algorithm for Twitter, can they have guessed what the echo chambers and filter bubbles will cause in the society and make all the radical approaches even more radical? Like that's so much in the future. But when these tools are becoming more and more available to people, it also changes the way certain things are done. So what are the ethical pitfalls there, like of this kind of technology? That I think is, I mean, for companion animals, we've been discussing this, right? I mean, you develop a simple robot to give your cat food when you're away. Will this, for example, make it easier on people who want to live the cat for a week, for example, with the robot at home? And will they actually end up damaging these pets' lives, right?
I would like to generalize and comment on that. Are ethics different for companion animals and sea life and bacteria that we saw? So if we discuss what is our common denominator here, when we work on such different species, I mean, even if we work on this animal ethics, what do we do about the diversity of the species?
Typically, you talk about harm, right? I mean, you try to balance benefits and harms, but I don't think anybody's trying to benefit harm balancing for bacteria. Right? I mean, for companion animals, we may do that, but also for maybe whales or interesting animals like tiger sharks or coral reefs. But are all sea animals or all animals as interesting? For example, can we build a citizen science project that will help cockroaches? I doubt that, right? I mean,
Okay, Helgi, would you like to add your perspective?
Yeah, we're still thinking about the scale, which is somewhat related then to the scale of the impact you have as well. I wonder what are the things where we have to work together as a community. I see this breadth for research and smaller groups doing something for bees and fish and what kind of things. And the platforms we create now, it's definitely the right direction. Do we need something else as well? Do we need to think in our algorithms more to make it more scalable? That's I think one path forward. But I don't know if the answer. It's one observation. What do we need to do is to work better as a group than individuals?
So I can report that most of the time you get the comment or you consider only one species that they apply to other people. To answer, if you want to get your paper published, we need to work on generic and so, but I agree on having data benchmarks. So with clear benchmarks, I think and this is naturally chances to consider it and with the contributions.
They say we should run some challenges.
Yeah, action items for the next year.
I would just love to say we should generate benchmarks, but we need to be very careful that we're generating them with the people who actually care about that species or that data, and with a lot of nuance and context. Because I know I've seen a lot of benchmarks, computer vision benchmarks for biological data, where it's just like you're memorizing it, like some version of you're memorizing it, and the model's not going to be useful. So I think we, particularly this community, we do want to think about how do you generate multi-species benchmarks? You know, how do you generate things where we're sort of trying to build, who knows, work towards something together? It should make sure that it's not like the aggregate at the detriment of any specific problem. And I don't really know if there's like a perfect way to do that, but just, we need to make sure the benchmarks are representative.
Yeah, there is a comment there.
I had a very specific question about synthetic data sets. We talked a lot about data optimization today, working with small data sets, or labeling very large data sets and how we can do that, as well as latent spaces, like these translational models, say like language vision or synthetic data in this real world. My question is synthetic data, is this ability to generate synthetic data, train translational models, is that something of shared interest in the community, or is that something that we expect a very like high effort, maybe lower support?
Anyone else? Any thoughts?
For the cases that I had, there's a very clear advantage, I think. How you would create synthetic data for the type of interesting deep sea creatures that we have seen.
Yeah, I think static data is great, but currently for what we have for humans, not and create static data set, static synthetic data set for each animal species. I think there is a paper here, CVPR, in Japanese, where they are so I think that's a very interesting action where you generate the synthetic data you need, a simple tool that everybody can use. I think that's very interesting.
I think it's very, very hard. We look for birds. And we talk to Pixar also. Most of the stuff is Pixar is really animated by hand. There's no really biomechanical process of the what size we can buy birds for $500. But these models are really very restrictive.
Okay, thank you. Thank you. Any comments? Yeah. Go ahead.
One more thing.
Yes.
One more thing.
So I was thinking, if you release a bird into the Panoptic Studio, maybe you can get a full 3D. So there are some generic tools that kind of might give you the kind of data that you can actually then crop scale put into your. So maybe we need some new ways of generating synthetic data, not just generative to sale networks.
Actually, we did the opposite. Never did that. I did it all the way.
I have just one more comment on the data set challenge aspect. I think it's really an opportunity for computer science to not have just squared errors. Maybe the task is like explain the mating behavior of the birds. That's your task. And maybe in between you predict some key points, but they need to be good enough to when do the task. So, but maybe you come up with some other engineering which is better for the task. So having this end goal as the challenge maybe is the simple part.
I have one more comment. I mean, this is a real pain, the CVPR discussion. With every student, people want to work on animals. It's painful as you can get. But even more painful for people like me who didn't grow up in this situation. But I have full understanding. I mean, I cannot start saying about this anymore. So I cannot go into psychology about this. So I think the long term may be in biology, or if this is generally in AI for science, this would go AlphaFold or something like that. 90% of AI for science has lower number of applications. That's why we have so many workshops. Also, I think what would really ease the situation is that biology departments hire more computer science PhDs, not biology PhDs with computer science work. That would make this at least attractive for people who want to follow an academic career. And also make more attractive also publishing journals and really follow the long-term painful science research to do that. Again, publishing at CVPR, which is painful in a different way. So that's and really cultivate. I think this is a matter of culture. And it is up to us also who write on Twitter, etc., to cultivate the importance of working for science. For example, tell your students to work, to go to work to NASA, for example, right? For PhD students whose dream was to work to NASA. Even now, one of my postdocs went to JPL. And really actually trying to promote science results. Like the AR glasses from up on there.
Yeah, I wanted to add to that today. And I'm not a domain expert, but the whole time I was listening to the talk, I had this recurring question in my head, like, what real world problem does this actually solve? And I think if you do that before you start on any of these problems, I think that's so important. Because there are whole communities, groups of researchers, so resource constrained that could not imagine what you are able to do with the algorithms that you've created. And you can be solving very timely real world problems with them if you make that connection. So please just start with answering that question first before diving deeply into things.
I feel like something that's been brought up in my experience as well is that, so I guess going off of this is that there are all these AI developments that we're coming up with computer vision. But in science, I feel like oftentimes it's not even the algorithm that's the bottleneck, just the interface to it. So I guess I'm curious about your perspective on where this human-computer interaction research between like bridging these two gaps fits into academia as a whole, and like how should we conduct it?
So Brent, you probably have something to say to that.
Yeah. So I think it's incredibly important. Yeah, I think that's kind of this like next phase of challenges that we should start thinking about. So get models that, you know, are working on our benchmarks and then try to get them in the hands of experts that can use them. The publication venues for that type of research may or may not be CVPR, but it certainly could be other venues like Chi or even Dub, or, you know, one of the knowledge communities. Like, yeah, I mean, I think we just need to be a little bit more open of like where we're going to be trying to send this work. You know, like, yeah, CVPR and these other NeurIPS and these other places are always going to be this kind of like challenge to get some of this more applied work into. And so I think we just need to be like more open to exploring some of these different conferences. But yeah, absolutely. I mean, I think that the challenge of how do you, and again, this kind of goes back to this trust, like, you know, I think we've built models that are useful, and now how do I convince someone like Akani that they are really useful? I mean, that's like both using the model and presenting the results in a way that she can understand, in a way that she can interact with. And then, I mean, the thing that gets me excited is like, often when you show someone like Akani these type of results, she's immediately wants to give you feedback, right? So like, how do you then take that feedback and actually, yeah, they're very easy to criticize, or quick to criticize, I should say. So like, yeah, how do you get that stuff back into the system and improve it? I think it's that next step of this progress, which is, it's exciting that we've gotten to that point, right? I mean, certainly 10 years ago, this was not the case. But yeah, now it's like we get to start thinking about these next set of challenges.
Yeah, I think actually this workshop is incredibly important for creating a venue where you can explore this kind of. So I think we should aim at making this conference, maybe like the Face and Gesture was a workshop.
Nice idea.
Yeah. Yeah, on that note, we're always open for more co-organizers to give you ideas. To run a challenge or whatever you think, and let us know, reach out. If you want to nominate somebody else too, who you think would be very suitable, send us the name, we will find them. So yeah, happy to grow.
Maybe we can start with an email list, kind of build community. That's, I think, always a good place to start.
Yeah, certainly we can do it.
Yeah, I will create an email list and put it on the CV4Animals webpage, so you can all register and connect further.
There's also the
Yeah.
AI
for conservation plans. Great.
Yeah, we can use that too. Maybe.
Yeah, go
maybe
for it.
there's more.
It's already 1,000 researchers plus globally working on these types of problems for animals. Usually in the context of conservation, but it's a growing community. So feel free to go on there and make yourself a subgen.
Okay, sounds good.
Yeah. Okay, on that optimistic note, we thank our panelists. Thank my wonderful co-organizers, and now we are headed to the posters. Thank you, and see you next year.
Yeah. Oh no, thank you so much for inviting me. Yeah. I'm not sure if I contributed anything, but yeah. No, that's very cool. I know it's always like I wish one of these was on like a different day. Like, yeah. Oh, thank you. No. It's awesome to be here. Sorry.